Got an excellent slate of talks for you this afternoon, but before we get to that, we got some housekeeping to do. First, I'll start by introducing our chairs who are joining us remotely. Jamie Elsela and James Lewis are the chairs for today's session. My name is Eric Parker, and I'll kick it over to our moderator to my left here.
Good afternoon, everyone. My name is Bailey Colburn, second-year PhD student at UNLV. Happy to be here.
Bailey, why don't you tell us a little bit about the session schedule and Q&A?
Sure. So we're going to be doing 4 talks at once. Each one will be 10 minutes. At the end of your 10 minutes, you'll see the timer at the bottom. You'll know you're done. If you do go over the 10 minutes, we'll stand up to let you know that you have reached your time limit. And then we'll be followed by a 20-minute Q&A. And if you do come up to ask a question, please come up to the microphone, introduce yourself, your affiliation, and then please address who you're asking your question to.
Okay, and then just a couple other things. Streaming and photography by audience members of the talks is kindly requested that you don't do that. And we also have one other reminder. There are student presentations in this session that have signed up for the Dwornik program that need judges. So if you are available to judge or serve as a judge in the Dwornik program, we ask that you please volunteer yourself. Every registered participant at the meeting should have gotten at least one email about this process for signing up. And so if you're interested, please look back at your emails. There's a simple link and login info, and you can identify the student presenters in this session to judge their presentations. So we have Jamie Elsela online, and we also we may have James here online. We're going to let our session chairs do the introductions for the speakers today. So let's see. Let me take a look at my screen here. Jamie, are you on
I'm
and available?
on. I think James might not be on the panelist link yet. So I just sent him a message, but I can take over until he gets on. But I think we have to wait a few more minutes before we start with the first talk, correct?
You're right. Yeah, we are about 3 minutes shy.
Okay.
All right, we'll get started very soon. All right, we are at the starting point now. We're going to start with the first talk. Jamie, would you mind introducing our first speaker for us?
All
Hey, Jamie,
right,
I'm here if you want me to step
Oh,
up.
go ahead,
Sorry,
James.
sorry,
You're
man.
on now.
There you go.
Sorry for the delay, everyone. So for our first talk is from Brian Hynek, and it's about colonization at a nascent hyperacidic hydrothermal setting relevant to early Earth and Mars.
All right, and thank you very much for the opportunity to be here and talk about a really cool natural laboratory where we can kind of look at what was happening on early Earth, early Mars, and the inhabitants in that. And I want to just note my collaborators, current and former students here. Justin has the next talk. Sydney has a poster tomorrow night, and then our in-country support that we have down there in Costa Rica. So let's take a field trip to Costa Rica. There's San Jose, the capital. Poas is about 9,000 feet high. It's an active basaltic andesitic stratovolcano and extremely low pH in this lake, down to -1.5. At times, temperature goes up to boiling, has frequent eruptions, has every state of sulfur, redox state of sulfur. And we began here studying it as a geochemical and a mineralogical analog. And then we did some microbiology sampling of this lake here. And we published in Astrobiology. This is the old stuff, but we found the biodiversity of this lake was a single organism, a single genus bacteria, acidophilium, that was living in that lake. And so for early Mars, early Earth, this is probably a very good analog because we had presumably very low biodiversity, low biomass. So we go and sample this lake. We work fast because there's magma down underneath the lake, and when those two interact, then you get these phreatic eruptions coming through the lake. And this is thanks to our USAID camera taking these images. So we were back there. That original microbe was there in 2013. Let's go 4 years later. It's a little more clement, but still extremely extreme. Did the microbes change? Did the community change? No, we found the exact same bacteria living in this pH 1 lake. And so since then, we've done shotgun metagenomics. This can help us look at how this thing can survive there, because when I touch the lake, my skin starts dissolving, but this microbe can just happily be there munching sulfur. And so how it can deal with the stressors as well as get energy. And what it also does, it produces these PHBs, these polyhydroxybutyrates. These are microbial plastics. It's generating plastics. And within those plastics are ATP. So it has all these energy reserves for times of harshness. So it would direct you to that paper there. And then we just a couple of months ago published this paper about these PHBs and how we should really consider these as a potential biosignature because they hold up a lot better than DNA in a Mars-like conditions. All right. So 5 days after our 2017 campaign, I was on the airplane and we had to deplane. We were getting rained on with ash. Went into major eruption. So it went from this quiescent, beautiful lake to this sort of hellish thing. The lake drained, it was gone. And yeah, it looked very different, but totally sterilized this crater with all the magmatic and volcanic events that occurred. So then come to 2019, the activity calmed down and the lake reformed. And so we were the first ones to come into this nascent sterile lake. And to our knowledge, no one has ever gone in and been able to document from the beginning a sterile aquatic environment here on our planet to do sort of studies of colonization and ecological succession of this new sterile lake. So a very cool opportunity, and really relating again to early Earth and early Mars, where we had much higher heat flow, lots of volcanism, magmatism, lots of impacts from above. So there are local to regional sterilization events in hot acidic environments, just like POAS, happening many times. So here's a cool modern example where we can sort of get to some of these big questions of early Earth and Mars. There's Justin, considering if he wants to still be my graduate student. A lot of sulfur down there. And here's the lake when we got there in 2019. We're trying to find a safe way down to that roiling, boiling, it's weird looking. It's just loaded with elemental sulfur. That's giving it the yellow color there. A lot of SO2 leading to sulfuric acid. Also a lot of hydrochloric acid in this lake. There's our in-country support. Definitely a gas mask type of a day, as it always is there. So what did we find in this brand new lake? Nothing. We found that it was totally sterile. It could not pull out any DNA from this newly formed lake. But ever since that time, we've been going back, either our collaborator or us, are there every few months. And since 2019, we have this long time series of samples. And so we can do this colonization study, which is really neat. All right. So yeah, here across the bottom, from left to right is time. So here's August of 2019 up through 2022. And these dots are all our sample days. And temperature is shown there in the red. So temperature range, about 50 C throughout this time. And I didn't plot pH, it just gets confusing, but it stayed right around 0, a little bit negative most of the time, which, yeah, it's more acidic than car battery acid. So this is like super, super acidic. And then in the green there are biology samples. And so on the scale bar on the right side, y-axis, these are DNA sequence reads. So this is sort of a proxy for biomass, let's say. And so these green dots, you can see starting on the left side, sterile, 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 sterile. And then we found just a bit of a microbe, a bacteria come in. Not the same thing we had seen before, which is what we expected. But this Diana caccata, these things are, they put the spores on the outside of ISS and then been able to revive them. These are super extreme microbes. But then they went away, and we're no longer in the lake the next time we sample. And instead, we saw a new bacteria that we hadn't seen there before, these sulfolobacillus. These things, optimal growth for these microbes is below pH 2. So, and they do a lot of sulfur oxidation. And then, but then they went away, and then found an archaea at this sample point, these thermoproteota. And these things, some have argued that this is the original archaea, where archaea branched off from bacteria. So all these things we're finding are deeply, deeply rooted in the tree of life, but none of them can persist, it seems. So we were there Thanksgiving 2013 or 2023, and all of a sudden there are all these hot springs flowing out of the crater wall into this lake. And so our main idea was that maybe these microbes are hiding in the underground connected groundwater system in the crater walls. And then when things are clement enough, they come into the lake. So these would be the sources. So that's our hypothesis, that these things are in an interconnected groundwater system, because no one really knows how stuff gets around from place to place. Here's a place we could test that. So here's a video from my drone. Some of these these springs coming in. The lake is right behind us. And so these are all hot environments. Most are around 90 C. And there's Justin in the steam there sampling. So we're getting these springs and what microbes are coming out of the springs. And then we want to compare that to what's in the lake to see if this is the source of these microbes coming into the lake. So here on the left are the stacked biology plots of the things that were coming out of the springs. And really, it's one thing. This is Sulfolobacillus, which is an archaea, sulfur eater. And all the springs had the same microbe coming out of them. So let's compare that to lake. What was in the lake? We couldn't get any DNA. So even though these springs with microbes are flowing into the lake, and we're sampling 20 meters down the shoreline, the lake pH was so extreme, we assume that these things are just dissolving, and we can't even recover even relic DNA from them. So that was a bust, our hypothesis. But to finish up here, just things can inhabit extremely, extremely harsh environments here on Earth. And, Mars had acid sulfate conditions and hydrothermal systems, but more like at pH 3 and not 4 orders of magnitude lower. So, that's good. Hopeful for Mars. And then what is trying to come in and colonize this lake are some of these really, really deeply rooted things, both archaea and a lot of bacteria coming in there. And we've detailed how they can deal with a lot of stress, how they can get their energy, and so on and so forth. And we showed that a single species, this original acidophilium bacteria, persisted in this lake over a 4-year time period until it went into major eruption. And then now we have this unique opportunity to do this colonization and ecological succession study. And so since that time, showing you the bottom here, sort of the summary of the microbes that have been in there. through time. And a lot of things have tried to come in and establish themselves, but after a month or two, a few months, then they're gone and no longer there. So none have been able to come in, take hold. We've never seen the original microbe, and they keep trying, and we still don't know where these things are coming from. Not from the springs, apparently. So just think early Earth and Mars had a lot of environments just like this. And so we think this is like a model ecosystem of low biomass, low biodiversity to try to understand the earliest conditions and how life got going and persisted on our planet. Thank you.
Okay, the next talk will be from Justin Wang, and it's titled Astrobiological Implications for the First Study of Microbial Life in the Hyperacidic Crater Lake of the Rincon de la Vieja volcano in Costa Rica.
Hi, everyone. My name is Justin Wang. I'm a medical student at the University of Southern California, formerly a graduate student at University of Colorado Boulder with Brian in his lab. So a bit about our lab, he talked a lot about our work at POAS. We really study extreme Martian analogs. So this includes Costa Rica, but we've also studied analogs in Iceland, Tanzania, and Argentina as well. I also want to do a shameless plug to Sydney. She's an undergrad in our lab, and she's doing a poster at tomorrow's astrobiology session. So highly recommend check it out. A big connection between all the sites we go to is really we're looking at places with low pHs, high temperatures, and this acid sulfate hydrothermal activity. Why acid sulfate hydrothermal activity? When you consider acid sulfate alteration minerals we find on Mars, specifically in Gale Crater and Gusev Crater, we're really trying to match that with our analogs here on Earth. In the Rincon de la Vieja volcano, which is my presentation today, we find gypsum and anhydrite, which we see a lot of in these environments. And so, so we're interested in acid sulfate alteration minerals. We're interested in the origins of life on Earth as well. And many of the theories of the origins of life on Earth are that life, the LUCA, the last universal common ancestors to life on Earth, likely came from a thermophile that was in a hydrothermal environment, either deep in the ocean, on the surface. And so really we're considering how could life on Earth have started, and how could life on Mars potentially start a similar way. We want to look at microbes in these kind of places. Another thing to note is that LUCA is predicted to have started forming maybe 4.2 billion years ago, early on in Earth's life, and potentially when it could have arisen on Mars as well. So a little bit about our environment, Rincon de la Vieja. So if you can see here in the map there, down smack in the middle is where POAS is. And then near the top there, top left, where the star, right here, that's POAS right there, and that's the star right here is where Rincon is. It is the only active volcano in the Guanacaste northern region of Costa Rica. The last time we sampled from this lake was in 2013, 2014. We didn't really look at microbes. It wasn't our lab, but they did mineralogy studies looking at pH was pretty much 0. Temperature was about 33 Celsius. The gas was relatively CO2 rich compared to the other volcanoes in Costa Rica. And right here, lots of sulfur, lots of chlorine, and mineral precipitates were dominated by gypsum and anhydrite. And so Rincon de la Vieja had its most recent magmatic eruption in 1967. This right here is from 2021. And so basically since 2011, there's been a lot of phreatic eruptions. And we thought this is a really extreme environment. We love extreme environments, and we should go sample from this. And so this happened in 2021. And we decided that within a year, we have to go back and see and test our hypothesis about extreme life, early life, and colonization. So within a year, we climbed up those lahar flows, very scared, but very exciting. And we went up to the crater and sampled from the lake. And so this was the lowest point we could get to the lake. So unlike POAS, where we were able to actually hike to the lake, here we cannot hike to the lake. It's like the closest point is 100 feet high off the crater wall. And so what we did is we got a water bottle and attached it to string and threw it over, which we call in my abstract the fishing technique. Basically, what we did is we had an Nalgene bottle. We did as much sterilization as we could. We put it in boiling water. We sprayed a ton of isopropyl alcohol on it the night before we went out to sample. We tied it to a rock and some string and threw it 100 feet off the wall and carried it up. And so the other thing we could have done was maybe a drone, but we didn't want to lose a $2,000 drone. So this was the best thing we came up with. When we brought up the sample, we measured temperature. We say greater than or equal to 38.4 C. The reason why is that it took us 5 minutes to carry it up, so it likely cooled on the way up. And then pH was less than or equal to 0. I don't know if you can see the colors here as well, but it was like immediately went to like the minimal like detection color, like super yellow. And then when we did ion chromatography, we also got like less than or equal to 0 as well. So, I mean, extreme acid. Yeah, and so we did fluid chemistry analysis with ion chromatography. We got a lot of sulfur, a lot of sulfate, a lot of chlorine, not as much as 2013 and 2014, but still a lot for environment and very extreme. And what we liked about this one in particular was that there was a lot more chlorine compared to POAS. So we think it's a little bit more representative of Mars and the chlorine that we find there. We did 16S rRNA gene sequencing and compared it with the Silver database. Very interesting enough, we had very minimal reads. We did a ton of, Sydney, the undergrad we work with, could talk about all the filtering and all the power max extractions we tried to do to get as much biomass as we could. We got pretty low reads overall, but what we did find was really fascinating. Most of the things belonging to the Dinococada phylum and Thermaceae family. So a little bit about that. So the Dinococada phylum hosts a lot of polyextremophiles, thermophiles as well, but what's really famous in this phylum is Deinococcus radiodurans. It's like in every single astrobiology textbook. It's really radiation resistant, really desiccation resistant, really good extremophile. In the phylum, in the family Thermaceae, we have lots of thermophiles. And this family is interesting, it's really deeply rooted in the phylogenetic tree as well. So basically, it's an ancient bacteria, as they say. And all of our reads belong to Thermus. And the Thermus bacteria, which we also find in POAS and we find in Laguna fumarolica, which is what tomorrow's poster is about, is famous for being in Yellowstone. It's actually what Taq polymerase comes out of. It's how we are able to do PCR. So it's a really well-studied bacteria that allows all of our lab reactions to work. It is found in temperatures from 30 to 80 Celsius and pHs from 5 to 10.5. So something that's interesting for us is that we have a pH of 0. So what we found is a way more extreme pH than most of what we can find for Thermus bacteria in literature. The next big find in our 16S sequencing is bacteria in the Bacilliaceae family. It includes aerobes, anaerobes. They are extremophiles as well. They can basically live through spore formation. And so that's how they're able to survive these extreme places. We see them when they've done swabs in the inside of the ISS to look at what's inside there. We've seen a lot of Bacilliaceae there. Some Bacilliaceae could be from contamination. I'll talk a little bit about that as well, because I know we used a water bottle. But majority of the reads we had actually belong to Geobacillus, which is a well-known thermophile that lives in hydrothermal environments. So we are hopeful and confident that likely what we have is local, what's native to Rincon de la Vieja. So a little bit about limitations. First is, our sterilization technique is not permanent. Truthfully, we weren't completely sure we were able to actually go just based on the danger. But that week there, we sampled for POAS, we checked Recon, and it looks really good. So we're able to go, and that's the best sterilization technique we could get. The other thing to consider is that, even if we sent in a bucket of yogurt and got something out, it's likely a lot of that would have been sterilized as well, just because of how extreme this environment is. Another limitation too is our rope method. So when it took us 5 minutes to get the sample out from the lake, it is likely that we collected some mud from the lake because we couldn't see the lake at the time we threw it in. We also maybe collected some dirt from the walls and got some soil microbes. It's also a possibility and something that we want to consider in our analyses. And lastly, so we only have a single time point here. So we've seen many different studies. We've had multiple time points, and only having one sample, we'd like to do more in the future. All right, so lastly, applications to Mars. What we see here is an environment with low biodiversity, but extremely complex bacteria. We've seen this in a POAS volcano before, What we also have here is bacteria with ancient bacterial lineages. So old bacteria, deeply rooted in a phylogenetic tree, which is exciting for considering what life on Mars, could have been like if we look at early life and the development of life on another planet. And then, yeah, I mean, our big, motto in our lab here is that we want to search for signs of life in these analogous environments. So really, we want to keep on emphasizing searching for life in hydrothermal environments on the red planet. Thank you.
The next talk is by Philandia Gaunt, and it's titled Biosignature Preservation in Acidic and Circumneutral Icelandic Hot Spring Deposits.
Green is forward. Hello, everyone. Thank you for that introduction. My name is Philandia. I am a PhD candidate at the University of Florida. And we're going to move away from Costa Rica and talk about a new site. We're going to talk about Iceland, and I'm excited to share with you my work on preservation, detection, and how this can impact future Mars exploration. If I could move my slides. A big green button, thank you. So we're all interested in Mars. We think it could be potential location for there to be life outside of our particular planet. And there are a few reasons. One, because there was a warmer environment. So whether you believe that it was cold and dry or wet and warm, we can agree that there had to have been at some point the presence of liquid water. There's also been a diversity of prebiotic molecules found, and there is an energy source available for metabolism. And one way to study Mars without leaving this planet is to use analogs. And I'm interested in Iceland. And so these three images show different hydrothermal systems that are on Mars that are very similar to Iceland. And the one that is most interesting is the middle, which is the Nili Patera that has hydrated minerals, which is something that we see in Iceland. And because Iceland is such a compelling target to look for biosignatures on Mars, there has been a few studies that have been done on it, looking at modern and ancient hydrothermal systems, looking at different textures, such as mud pots and hot springs and fumaroles, but no such study exists to compare acidic to circumneutral systems, looking at preservation, detectability, and also detectability with modern spaceflight technology. So to answer that question, I have two sites in Iceland. One is acidic and one is neutral. The acidic system has a pH of 4.05 and the neutral system has a pH of 7. And if we take a closer look at these two samples, the acidic samples 1 and 2 are closer to the source and the rest are further out. It goes from A1 to A7. I also have total carbon in mineralogy for each of the samples. And so the highest is 0.3%, with the lowest being 0.09. The majority of the mineralogy is goethite and quartz, but I also highlighted the sulfate that is found in A3. I think that would be a very interesting mineralogy to discuss. And then in the circumneutral, there are 3 sites, N1, N2, and 3, with N1 being closest to the vent. And you can see the total carbon is much higher than the acidic. It's 0.58 to 0.41. There's also the presence of the calcite and the opal A and that amorphous silica. So to look for biosignatures inside of the rocks, I use a method called gas chromatography mass spectrometry, or GCMS. And to do that, there are 3 main ways that you can operate the instrument. So you can do pyrolysis, which uses heat to break down larger molecules into smaller molecules. You can use derivatization, which uses a reagent to make organic molecules less polar, which then makes them more detectable using GCMS. And then you can also do thermochemolysis, which degrades organic compounds through heating in the presence of a chemical reagent. And so I have four main methodologies. First, using neat flash pyrolysis, neat meaning that there is no reagent. And flash pyrolysis means you put the sample in an oven that's already at that temperature. So the two temperatures that I used were 600 and 800 degrees Celsius. And so there are two thermochemilysis reagents that I was interested in, tetramethyl ammonium hydroxide, TMAH, or trimethyl sulfonium hydroxide, TMSH. And they work in similar ways in that there is this Lapol hydrogen in the carboxylic acid that the thermochemilysis reagent works with to make a methylated carboxylic acid that is then detectable with GCMS. And then the last method is the ramped TMAH, which is similar to what is currently located on Mars. NASA's Curiosity rover has a GCMS on board, its Sample Analysis at Mars, or SAM instrument, and it ramps up at a rate of 35 C per minute. So now we can step through the results. This shows a graph, and on the x-axis is the acidic samples, A1 through A7, and the neutral samples, N1 to N3, and then the number of organics detected is on the y-axis. So for pyrolysis, I was looking for alkanes and alkenes, and you can see that there are none detected in the acidic samples. There are some detected in the neutral samples, but there are more alkanes and alkenes detected using the pyrolysis temperature at 850. So from this, we learned that temperature alone is not enough to break down bonds in acidic samples, and also that the pyrolysis temperature of 850 is superior to 600. So moving on to our first reagent, TMAH, we would be looking for FAMEs, and you can see in both the acidic and the neutral, there are a host of FAMEs that are detected. But you can also look for alkanes and alkenes, and you can see here that in the acidic sample, there were more alkanes and alkenes detected than the neutral, but they are both present. So from this, we've learned that TMAH is a great reagent for methylating both fatty acids and also alkanes and alkenes. And so if we move ahead and if we look at TMSH, you can see in the acidic sample, it's not really anything present. There's that 1 in A3, but there are more FAMEs detected in the neutral sample, but they are less than what we saw using TMSH. And then also looking for alkanes and alkenes, A3 still has a little something, but mostly the acidic doesn't show anything and the neutral system does. So from that, we see that TMSH is less efficient at methylating organics in acidic conditions than TMAH. And finally, if we look at the ramped pyrolysis, which is similar to what we see with SAM, there are no FAMEs detected in the acidic sample, and there are a few FAMEs less than with straight TMAH, but there are a few FAMEs detected. So from that, we learned that TMAH ramped pyrolysis is capable of liberating organics in the neutral conditions. So if we were to sum this up, we can see that total organic carbon and mineralogy are important for preservation, but my results show that there are other factors that can enhance detection in biosignatures. So neat versus reagent, making sure that you're using a thermochemilysis reagent, TMAH versus TMSH. TMAH is superior to TMSH. You can look for both fatty acid and alkanes in either acidic or circumneutral. And it's also important to use flash pyrolysis instead of ramp pyrolysis. So I've talked about preservation, I've talked about detection, and now I'm interested in preservation. If I take all of my samples and separate them by mineralogy, there are 4 main categories that arrive. There's the goethite, and in goethite we saw there were acyclic hydrocarbons present, and so that indicates that it might be a good mineralogy for preserving organics, so that would be new information. But if we look at carbonates in the silica and the morphosilica, we see, especially in the silica, FAMEs, acyclic hydrocarbons, and that would be no surprise. That's well cited in the literature that that is a great mineralogy for preserving biosignatures. And so the last one is the sulfate category. I just had one sample from sulfates, and we were able to see FAMEs, but not any of the acyclic hydrocarbons. And so I would like to do more work, expand my sample set to get more data before I can really comment on sulfate. And so how can we put all of this information together. So the future of Mars research, one such rover is the Rosalind Franklin rover. Through ESA, they have a GCMS that's going to be on board as well. And so it's landing in Oxyoplanum, and this is an image there that shows the hydrated minerals that are present, and then the red tracks are where the rover will be roving. And so I haven't been able to find any information about whether it'll be acidic or neutral, but my results show that it doesn't matter if it's acidic or neutral as long as we use the right procedure. So as long as we're using flash pyrolysis with TMAH, we will be able to identify a diversity of biosignatures inside of Oxyoplanum. So with that, I would like to thank my PI and the team that collected the samples, and I look forward to answering your questions later. Thank you.
Okay, the final talk before the first Q&A session is from Carolyn Harris, and it's titled Characterizing Lipid and Isotopic Biosignatures in Terrestrial Hot Springs with Relevance to the Search for Life on Mars.
All right, thank you so much for that introduction. So the research I'm presenting today is part of my dissertation research at Dartmouth. Thanks to my co-presenters for setting up the relevance of hydrothermal activity to astrobiology so well. So first, hydrothermal activity is linked to planet formation and evolution, and Mars is likely to have experienced hydrothermal activity during its early history from volcanism or large impacts. This is important for life because it perpetuates regional hydrological cycling and can provide a source of water in an otherwise arid environment. It also creates lasting physicochemical gradients that can be exploited by microbial life. This is one of the reasons why life on Earth is thought to have originated potentially in subaerial hot springs. And finally, these sites have very high mineralization rates, and so they can entomb and preserve evidence of their microbial inhabitants in the form of both morphological and molecular biosignatures. And there's evidence of several hydrothermally influenced deposits on Mars that were likely active during the Noachian. And so because of this, modern hot springs are a valuable analog that warrant further study with respect to their microbial inhabitants and biosignatures. So while there's several or many prokaryotes that can live in acidic hot springs, archaea are the best at thriving in the hottest and most acidic regions. And this is in large part due to the unique cell membranes that they synthesize. So unlike bacteria, archaea form a membrane spanning tetraether membrane, which is a monolayer that is able to have maintain permeability and rigidity in the face of heat and acid stress. And in particular, many archaea produce a class of lipids called isoprenoid GDGTs. And so these are membrane-spanning lipids that can contain between 0 to 8 rings. And the more rings a lipid has, the more tightly that membrane packs, allowing it to withstand these extreme conditions. And so because of that, I'm focused on archaeal lipids as a biosignature. And so we can look at the 6 classes of biosignatures identified by the Mars 2020 team, and archaeal lipids are relevant to several. First, organics. So the lipids themselves can be well preserved in sinter deposits, and they're unlikely to be created abiotically because they have really complex non-random branching. They're also a very robust molecule that's likely to survive long-term storage on Mars, chemically unchanged. We can further constrain their biogenicity by analyzing the stable isotope composition of these molecules. Metabolic processes tend to concentrate the lighter isotope in biomass, and when you analyze a specific compound, such as a lipid, you can more tightly constrain biogenicity. And so while a lot of previous research has focused on carbon isotopes, today I'm going to talk about hydrogen isotopes. So some recent culture studies done on archaea have shown that archaeal lipids are uniformly depleted in deuterium relative to their source waters, representing a kinetic biologic fractionation and indicating their potential as a biogenicity indicator. So the goals of this study were to determine the geochemical controls on archaeal lipid production in modern hot springs to guide the search for fossil biomarkers in sinters on Mars, And we looked at three aspects of our archaeolipids. The first is just the concentration of lipids in spring sediments. The second is the distribution of IGDGT structures. And then finally, we measure the hydrogen isotope composition of these lipids, which represents the first measurements from a Mars analog environment. And so to do this, we kind of sampled two broad regions of springs. The first is Yellowstone, which has extensive geysers and hot springs that can be acidic to alkaline. And then we also sampled a handful of hot springs from the El Tatio geyser field in the Atacama Desert in Chile, which is known for being a very robust analog for hydrothermal activity on Mars, in large part due to the hyperaridity of the region and the hypersaline waters. So super briefly, at each hot spring we visited, we did a YSI cast to determine the major geochemical parameters of the water. We collected sinter-rich sediments. We did some spectroscopy to determine the major ions. And then we did some organic chemistry to extract archaeal IGDGT lipids and to isolate the intact polar lipid fraction, which represents living or recently dead biomass. And then finally, we identified and quantified the lipid structures and then determined their hydrogen isotope composition via GC pyrolysis IRMS. So getting into the results, this plot is summarizing the pH and temperature conditions of the nearly 50 springs that we sampled for this study. And so these represent acidic to alkaline pH and warm to temperatures that approach the boiling point of each system. And these springs also span a wide range of other geochemical conditions, which will provide a host of explanatory variables to kind of explain our lipid results. And so in particular, I want to call out that El Tatio, shown in pink, has higher conductivity than Yellowstone, reflecting those hyperarid, hypersaline conditions. And also of note is that the water hydrogen isotope composition varies between the two regions, which is reflecting regional hydrological cycling. So we detected archaeal IGDGTs in all hot springs. This is the same temperature versus pH plot where the size of the points is scaled to the lipid concentration. And when we compared the log lipid concentration, which does vary widely, it spans 4 orders of magnitude, to our geochemical controls, we found that there's no dominant geochemical control, but this range is similar to the range reported for other spring systems. Next, we looked at the distribution of GDGT structures that contain between 0 and 8 rings. So this plot is just showing every spring in our study arranged by pH from most acidic to most alkaline. And you can see that the more cyclized lipids, shown in the pinker colors, are more abundant in the more acidic springs. And so we can quantify this by calculating the ring index, which is a measure of mean cyclization in a distribution. And when we compare this to our suite of potential geochemical predictors, we find that pH is the dominant control, and temperature actually doesn't play that big of a role. And finally, we looked at the hydrogen isotope composition of these lipids. And so this required an extra chemistry step in the lab in order to get a molecule small enough to analyze on our IRMS. We chemically cleaved the ends of these GDGT lipids to release the biphytane chains. So in this example, GDGT3 would be broken down into a biphytane 1 and 2. And then we measured the hydrogen isotope composition of those individual biphytanes. I don't have time to get into the kind of biphytane level differences. So today I'm just going to show you an abundance weighted average across all of the biphytanes. So this plot is showing the mean hydrogen isotope composition of all the biphytanes for a smaller subset of springs that we analyzed from El Tatio on the left and Yellowstone on the right-hand side. And so there is a little bit of variability, and in general, the Yellowstone samples are more depleted relative to the El Tatio samples. But these measurements are really best interpreted relative to the hydrogen source, which in this case is spring water. So when we add the spring waters to this plot, we can calculate a offset or epsilon, which represents the biological fractionation when an archaea incorporates hydrogen from water into its lipids. And this plot is showing that mean epsilon value or fractionation. And what we see is that in all cases, the lipids are between 150 to 250 per mil depleted relative to their source waters. And this is a similar variability to that seen in culturing studies on archaea. So this plot is summarizing all of the culture studies to date, which have been broken apart by the metabolism the archaea is doing, shown autotrophs and heterotrophs in aerobic and anaerobic conditions. And of particular note, shown in red, is measurements that I recently made on a sulfolobus strain that was isolated from a Yellowstone hot spring. And so we see that the variability among our springs is similar to that we're seeing in culture studies. And these differences in the fractionation are likely reflecting differences in central metabolism or the substrate used for growth, and not reflecting differences or being influenced by the geochemistry of a system. And so that's good if our goal is to use this as a biogenicity test. And so this consistent depletion it does indicate that this hydrogen isotope composition may indicate biogenicity. And so when we think about applications to Mars, the water in hydrated siliceous sinters would provide a background against which to compare a putative biosignature.
All right, if you could wrap up, that'd be great. Thank you.
Okay, yes. So just to summarize, we have found lipids from polyextremophilic archaea in all of the springs we sampled. Spring pH was the dominant control in cyclization. And most important conclusion is that all lipids are depleted in deuterium relative to source waters, providing further support to examine Martian hydrothermal deposits. And we've outlined an interpretive framework against which to compare the hydrogen isotope composition of putative biosignatures. Thanks.
All right, we'd like to invite all of our speakers from this first portion of the session to come back up to the stage for the Q&A session. And just as a friendly reminder to the audience, if you could come forward to the microphones to ask your questions, state your name, your affiliation, and let one of our speakers know who you're directing your question to. Thank you.
Hi, everyone. Bonnie Tace, JPL. I have a question for Flindia. I so enjoyed your talk and the structure, and especially the linking for the different reagents and the mineralogy. And I think at the start, you talked about the positions that you take in the samples in the hot spring fields. I wondered if you noticed any trends with the preservation related to the position within the hot spring itself, or was it random?
That is a great question that I had not thought about. I would love to go back and look at that. I think especially in the acidic system, there just really wasn't a lot of material to work with, but I could definitely comment on that with the alkanes and alkenes, and I will get back to you. And I would like to call out, you asked me a question about pore space last time. And so I think it's really interesting to think about how some of the samples had the mineralogy that was the quartz versus the opal A, so that like recrystallization really did not work in like preserving the biosignature. So I think like the larger pore space of the amorphous silica was better.
That's so cool. Thank you. 2 for 1.
Hi, Joey Pasterski from NASA Goddard. Bonnie just stole my question. No, not really. I had a question for Carrie. That was very cool work. And I wanted to know if you could expand out from just archaea, because you're looking at GDGTs, but GDGTs are also isoprenoids. So maybe there's some essentially what I'm catching from your work as a non-biologist is that the enzymatic pathways to build GDGTs are a little bit agnostic to pH. So then could you build out and relate the enzymatic pathways that archaea are using to build the GDGs in these hot springs and relate that to other types of organisms that might also reflect the same kind of hydrogen fractionation?
Yeah, that's a great question. So what we're seeing really is that in bacteria, the metabolism that the bacteria is using really overprints that environmental water signal. So this works best for autotrophs or for archaea that are using kind of similar pathways to synthesize their lipids, regardless of the metabolism that they're using. So all the data I showed were for phytanes or biphytanes from archaea. But yeah, that's a good question. And so bacteria that use similar pathways also show this characteristic depletion, but ones that use other pathways, they can be all over the map, like more enriched relative to source waters even.
It's still pretty powerful. That's powerful that you can apply it to other organisms also, though. Yeah, awesome. Thanks.
Thanks.
Hi, Guillaume Masson from Paris-Saclay University. My question will be for Felindia. First of all, very good talk. Thank you very much. And I was just wondering if for your TMAH and TMSH experiment, you were able to recover the same FAMEs, or did you observe some differences in terms of chemical structure for each one?
Yes. So there were a host of different FAMEs from C8 to I think the highest one was like C24 in that N2 sample. So there were some that were the same, but there are a lot of individual unique FAMEs that were found as well.
Mm-hmm. And do you have any idea of like which sort of chemical structure would work best for TMAH and or TMSH?
I will say that I think the FAMEs were better at that. So maybe that straight chains are better. Is that what you mean? Like straight chains?
Yeah, I mean like
aromatic?
saturation or branched or side.
Gosh, no, I haven't thought about that. I'll have to look into that. Thank you.
Okay, thanks.
Hi, Carl Allen, NASA JSC, retired. First reaction to particularly the first two talks is thank God for graduate students who will actually go into these environments for their science. The first samples from Mars that are going to be analyzed in a lab for this sort of thing are being collected right now. If we believe the NASA headquarters briefing from the last hour, the Mars samples might be back in 10 years. These are near-surface samples that have had a lot of radiation and other insults, so they probably don't have living things in them. But how would you recommend analyzing Mars samples in the lab for biosignatures, and particularly thinking about the work the last speaker has been doing?
What do you start biosignatures? Published on
Yeah. Yeah, I mean, that's more of like more of our stuff, you know, is DNA sequencing. If we're considering, yeah, I mean, the things that we're getting from Mars now, I mean, I would say like lipids is probably one of the best things, the longest living things. So I would give this to you with lipids. Yeah.
Thanks. Yeah, I would agree. I would say go for lipids, which are one of the longer lived biologic molecules on Earth. And then also, applying some of these techniques that we can do on Earth that rovers can't do, like nanosims, to really look at the micro level of isotope fractionations. I talked about hydrogen, but that really will be most powerful when it's combined with other isotope techniques like carbon. So you're getting a kind of multi-proxy analysis of biogenicity. Thanks.
We do quickly have one question online, and we also want to encourage the online attendees to feel free to ask questions. The question online is for the last speaker. They want to know how much biphytane in moles is needed to make the DH measurements.
That's a great question. It's 150 nanograms per analyte, so per individual biphytane.
Steve Ruff, Arizona State. For both Felindia and the last speaker, didn't catch your name.
Carolyn.
Carolyn. So I didn't quite understand the nature of the sample material that you both collected in your different locations. Is this actually chunks of sinter rock that you're collecting? You said sediment-rich sinter. I don't know what that, or sinter-rich sediment. So Felindia, where were you collecting this material and how did you actually collect it?
So I wasn't a part of the team that collected it, but I've seen it, and they were actual just chunks of rock. They were rocks that we ground up into smaller particles to put in the GCMS.
Okay.
I feel like you still have a question.
Well, and
Oh, okay.
Yes. Yeah. So we're calling it sinter-rich sediment. So we, used a pole with a sterilized beaker on the end to scoop sediments out of the margins of spring vents, which kind of depended a bit on the spring itself, but had like chunks of sinter that were, you know, kind of thin crusts to maybe like marble-sized pieces, which were then crushed before we extracted lipids.
Okay, so I guess I'll make a comment that, you know, this kind of work is both, it's the right way to start, but at some point you should go to literally places where there are extinct hot springs, and what is the preservation potential of this wonderful stuff you're finding in these active environments, right? But on Mars, I assure you, there won't be active environments, you know, so it won't be as diagenetically modified as typically hot springs are over time on Earth. We know from Spirit, for example, that it's still amorphous opal and silica, so it's not dramatically diagenetically altered, but it's still ancient stuff. And so what is the preservation potential? So hopefully all of your work will go in that direction. Thanks.
A great note. Thank you, Steve.
Hi, Gray Joni, Towson University. I had a question for the first speaker. Your work with the caldera lakes as a like analog for early life and its formation is really interesting. And if I remember correctly, I think Schmidt Ocean Institute did a expedition a few years ago where they found some chemosynthetic communities living like at least 2 kilometers underneath the vents in the crust. And I was wondering, do you think these caldera lakes could have a similar setup where rather than getting like replenished by other hot springs, it's just deeper down? And would you be kind of working with that in the future with your work?
I can't see you, so I'm going to say it's a bad placement here. Yeah, so let me see if I'm getting this right. Things a couple kilometers under the ocean, some microbes there. Is this like a surface expression of that sort of community? Is that what you're asking? Or how would we test if it's similar? Is that?
Yeah, like if it's something similar is happening with the terrestrial lakes.
Yeah, I don't know. I have to think about that, but there are a lot of differences, I think, because we have atmosphere and stuff being blown in. And so all these spores floating around can try. There's groundwater coming up. It's connected to other volcanoes. So yeah, I'm not sure. There are some significant differences, but also you find these extremely low biodiversity, you know, very, very specialized microbes doing their thing down there. So good question. Let's
And
get
then
more.
if I can like give another question off of that,
Yeah.
do you think maybe the reason that some of these caldera environments aren't like sticking around for longer than a few weeks or months is because of that atmospheric and like particle interference versus the marine ones where they're kind of more protected in a way?
I think it would be more because of the pH.
Okay.
It's just so acidic. I mean, right? Most lakes, you can find relic DNA from 10,000 years ago, 100,000 years ago. This stuff, it's like gone instantly when it touches the lake, unless it's adapted to survive there. So I think, yeah, it's more of a pH thing.
Okay. Thank you.
Yeah, that's my guess.
Can I say something?
Oh, yeah.
Yeah, I would also point you to Karen Lloyd. She does some studies with using hot springs as proxies for what's deep. So look into her studies, and she has some good work on that. Yeah.
Hi, I'm Jung from Umeå University in Sweden. I have a question for the first and maybe the second speaker. So have you ever detected any smaller unit of the DNA or biosignature because you detected some kind of microbe, but no biosignature in the spring, right?
Yeah. So I mean, all we've been able to do is look for DNA. We tried to culture them, but those were really difficult in the lab. It was very difficult in the lab, but we haven't been able to do just based off of like the chemistry. Like we haven't been able to like look for specific biosignatures. I did a study on using bioplastics as a looking for that on Mars potentially as a biosignature. but that's not something we've been able to actually like find ourselves in our actual like pH 0 samples.
But maybe it's good also look at the product of the decomposition as well. And maybe you can also do some experimental in the lab to see the kinetic and which product that is produced from the DNA from the water.
Yeah.
It would be very interesting.
Yeah.
And I have a second question maybe for all of you.
Mhm.
So you found some microbe, but it's really hard to find DNA in the low pH water or biosignature. So how do we develop a strategy to or a plan to look for biosignature on Mars? Imagine if there was microbe, but how can we detect that biosignature now?
Yeah, so for our studies is what we have is we look for DNA and we've like sequenced the DNA and that's what we have there. But on Mars, the difficult thing about DNA specifically is that over time with radiation, it degrades and doesn't live as long. And so that's why we were saying earlier that lipids is something that can live like way billions of years longer. And that's basically like some of our best biosignatures that are well preserved for this length. Do you have any guys? No? Yeah. Okay, great. Thanks.
Hello, Sophie Figuera from Carthage College. I have a question for Brian and Justin. I'm not as familiar with PCR off the top of my head right now, but you were talking about how the DNA degrades very quickly when it comes in contact with the acid, and I was wondering if it's possible that there is an adaptation or a modification to the DNA that helps stabilize it, but also makes it more difficult to detect with our current methods.
Yeah, that's a good question. So, I mean, with our current methods, there's many different theories of why, it's hard to detect some DNA there. Some of it is just the reagents we have aren't made for pH of 0. So there could be issues with actually like trying to clean, the microbe products to isolate specific DNA. Can you say the statement about you asked about PCR in specific or?
Just that I'm not specifically PCR,
Yeah.
just that I know that that's a method that is used in sequencing.
Yeah, for sure. Yeah, basically just like in the actual extraction method from like soil water sample to the DNA, there's definitely a lot of issues with, potentially the acid is degrading the DNA and potentially just what reactions we have are able to do just don't work well. So, and Brian, yeah.
Yeah, I can add a little too. So, and we do a lot of things like DNA attaches to gypsum really well. So you gotta like wash it off the gypsum. So we do things like that. Same with iron, some iron things. And then, I mean, even like with fluid samples, you normally want a lot of fluid, right? So you filter it. You bring a pump and you filter it. And so I did that, ran it through these Stereovex, 0.1 micron filters, and I go back and I have no DNA, but then in just my Falcon tube, I have DNA. And I'm like, I call the company. I'm like, what's going on? I sent 10 liters of fluid through this. And they're like, oh, these filters that below pH 1, they just dissolve. So you just dissolve the filter and all the DNA just washed right through. So we've had to do a lot of adaptations to try to even get anything out of these systems. And we're still, revising and learning as we go, but they're really, really hard systems to study.
Thank you.
Yeah.
Hi, Jordan Chastain, Caltech. Thank you all so much for your amazing talks. I have a quick question for Brian. So you talked about how your lake specifically went through like a complete sterilization and it dried out. There have been some papers about Mars also possibly having like wet dry cycles in the crater lakes. And I was wondering if you might have some ideas about how your system could relate, either compare or contrast based on the timing of those cycles, to systems we might expect to have occurred on Mars.
Yeah, good question. Wetting and drying. Well, Mars, we don't know the time scales at all, right? We just guess. But you figure if you had a plume source and these volcanoes erupted off and on over billions of years, a single volcano, right? So it was hot for a long time. And I would think you would have had long-lived in the crust hydrothermal systems that probably didn't really dry and wet. The surface fumarolic environments, yeah, those are tricky because those move around so fast and are just so active. And we studied a lot of those too, but the lakes are kind of more fun, I think. But yeah, time scale wise, I don't know. So right, this it had been like 50 years since a big eruption at POAS. Then it went into 2 full years of just nonstop actual eruptions. Then it got calm. And so I booked a ticket for 10 days from now to go to POAS. And guess what? 4 days ago, it started erupting. So I think our sampling trip is canceled for the end of March. But it's just super dynamic environment. And I mean, hot springs and hydrothermal environments just generally are. And so that's another challenge to think about both for Earth and Mars. So I'm not sure I answered your question, but a little insight and context what's going on at POAS.
Cool, thank you.
Thanks, Fred.
Thank you, presenters, and thank you everyone for asking your questions. That will complete the first Q&A. And I will hand it over to Jamie to introduce the next speaker.
Thanks to those great speakers. That was a great first set of talks. And so we're on to our second set of 4 talks. And the first one will be presented by Amy Williams. I believe she's presenting virtually. And the topic is Organics Detection in Ancient Cratonic Rocks with TMAH Wet Chemistry.
All right, excellent. Thanks, Jamie. And thanks everyone for coming to the talk. And I'm really sorry I couldn't be there in person. So let's go ahead to the next slide. You often hear about my work in the context of TMAH with the SAM instrument on board the Curiosity rover. Today, we're going to be talking more about what happens when you apply this to terrestrial rocks that are of similar ages to those on Mars. But let's go ahead and start with the SAM instrument first. So SAM is a GCMS. We have a quadrupole mass spec. We have a tunable laser spectrometer. And it's relevant to this study because one of the things SAM can do is detect organic carbon with pyrolysis GCMS with or without reagents. So let's go ahead to the next slide. And so wet chemistry experiments on SAM, what do they include? So the sample organics can be made more volatile and detectable to GCMS using either MTBS-TFA derivatization, which you see the reaction for in the bottom left, or TMAH thermochemolysis, which you see in the center left. And thankfully, Felindia already briefed us on TMAH. Next slide. So thermochemolysis, of course, is the degradation of organic compounds through heating in the presence of a reagent like TMAH. And we actually have 9 sample cups on board the SAM instrument that contain these reagents. So of all of the sample cups, 9 of them contain reagents and only 2 of them contain TMAH. So we performed the first TMAH experiment on Mars. We are optimizing for the second. And I want to tell you about some of the things we might expect to see if those rocks behave like ancient rocks on Earth. Next slide. So why use TMAH? It is really good at releasing polar molecules bound within macromolecules. So from the biologic perspective, this could be fatty acids within a cellular membrane or monomers from kerogen or macromolecular carbon. And so the TMAH will react with the labile hydrogen, for example, on the fatty acids shown in the chromatogram here on the right-hand side. When you don't have that reaction, your fatty acids are not really detectable to GCMS. But when you perform this reaction, of course, then you have the happy green check mark and you can see your fatty acids. Next slide. So the map you're looking at here is showing the locations of Precambrian and Archean, or Precambrian crust and Archean cratons on Earth. The Archean cratons are in dark gray, and those are going to be units that are greater than about 2.5 billion years old. The yellow dots are showing you where rocks greater than 3.6 billion years old have been detected. So as you can see, rocks of such extreme age are rare on Earth. But 50% of Mars's surface is putatively older than 3.6 billion years old. And so studying Mars, of course, is this great opportunity to get a window into a planet in its infancy. And of course, if life arose on Mars, that time frame is sort of the range we'd want to look in for biosignatures. Next slide. So in much of the ancient Cratonic literature, Archean cherts are sort of a focal point. Archean cherts are known to contain putative biosignatures in the form of carbonaceous matter that can be embedded in the silica matrix. Next slide. And insoluble organic matter, so kerogen, that's isolated within Archean cherts is likely the same age as the host rock. So the intent is that organics preserved are native to when they actually, or native to what was present when the rocks were forming on the order of billions of years ago. Next slide. And so this study assesses how TMAH thermochemolysis performs on some of Earth's oldest rocks. Let me step you through the sample suite that we use. So next slide. The first is a chert from the 1.1 billion year old Paradise Creek Formation. Next. The second is a shale from the 1.6 billion year old Barney Creek Formation. So these are both within the Precambrian crust. Now we're actually go to the next slide. I think about one more that should just be not quite on the craton there, but it's the 1.9 billion year old Beobic Iron Formation. It's actually a chert from that unit. Next slide. And then we get into the really ancient stuff. So we have several samples from the Pilbara Craton, ranging from 2.6 to 3.2 billion years old. This includes cherts, siltstone shale, and an arenite sandstone. Next slide. And our final sample suite is from the 3.4 billion year old Kronberg Formation. It's a chert from the Capval Craton. Next slide. So a brief note about rock processing. These samples were collected in the 80s and 90s. They were not collected with the attempt to be organically clean. And then they were subsequently stored in plastic bags in Dorothy's garage for a while. So this is not a really great start. But let me step you through what we tried to do to be able to isolate any organics that were preserved within the rock. So they were shipped to UF in 2019. We broke off interior gravel-sized pieces and sonicated them in solvents, methanol and dichloromethane, until the solvents were clear after sonication. We used an ash silica gel blank in our stainless steel ball mill as our background blank for anything that was in there during sample crushing. And then we solvent rinsed the ball mill and powdered the sample in it, and then collected the samples for analysis. Next slide. So we'll finally get into some busy charts here. So what we wanted to do was think about what would the SAM instrument be able to see if you analyzed rocks of this age. So what you're looking at is an evolved gas analysis suite of samples from all of these rocks. So starting with the youngest at 1.1 billion years, the Paradise Creek on the top left, and then down each column getting older till you get to the 3.4 billion year old rocks. And so in general, what you're seeing is the cherts really didn't do a good job of evolving organic carbon using this profile. Organic carbon evolved above 600 C in most shales and sandstones. And the organic matter, of course, was low in all of these cherts, at least as revealed by EGA, except for the 3.2 billion year old Sulphur Springs chert. Next slide. So now we're going to get into GCMS. So we actually performed TMAH thermochemolysis, and you can extract ions that reveal acyclic hydrocarbons or fatty acid methyl esters. So in this case, we're looking at the alkanes. And so what you'll notice is alkanes are present in all of these samples, and these are actually silica blank background subtracted. So this means that this shouldn't be from our processing. So it's possible it's from kerogen cracking as a result of the TMAH processing, which is exciting for releasing kerogen from these rocks. Next slide. And then finally, looking at the fatty acid methyl ester signal, so you'll notice that every one of these samples, the y-axis is scaled the same for all of them, but they all have FAMEs. FAMEs are not expected to be preserved over deep geologic time. So the signal is almost certainly from modern microbes or the collection and storage contamination that these rocks experience. But we can use this to inform ourselves on what the organic matter or what the native organic matter may be. Next slide. So what we'll do is compare with a study from 2008 and 2012. So kerogen can survive over deep time, but its biological origin can be controversial. In the bottom figure here, you're seeing an analysis using an HF-HCl extracted kerogen from an Archean chert. And the interpretation of those data was that it was a signal of biosynthesis. Next chart. So analysis in 2012 was performed on 4 additional Archean cherts, and the alkane signature was actually quite different from the original study. So TMAH thermochemolysis was performed to assess, why this may be, why there may be a difference. And they actually determined that decarboxylation of FAMES from contamination was likely the source for the N-alkanes they found in the 2012 work, but it's inconsistent with what was seen in the 2008 work. Next slide. So we know that FAMES are not expected to be preserved on their own in deep geologic time, maybe on a few millions of years, but not billions of years. And first, for our FAMES, our work is consistent with the 2012 work that our FAME signal is from modern contamination. Next slide. But if you look at our alkane data, we can comment on the aliphatics that come out of these rocks. So the aliphatic fraction of carbon can vary greatly between cherts even of similar ages. And what we see in our data are actually doublets of alkanes and alkenes, suggesting that some of the alkanes that we've seen are due to kerogen cracking, likely due to our TMAH hydrolysis at elevated temperatures. Next slide. So leaving you with the conclusions here, cherts are known to be ideal for long-term preservation of morphologic and benchtop extractable organic biosignatures, but no 2 cherts are the same. Other ancient lithologies, such as shales, are better suited for organics extraction via pyrolysis GCMS, plus or minus any reagents you may want to use. And so we'd suggest that Mars missions searching for signs of life with any SAM-like GCMS instrument that's using sort of an online procedure should target clay-sized grains within sedimentary deposits. So think about mudstones, claystones, and shales. And I will leave it there. Thank you.
Great. Thank you so much, Amy. Our next speaker is James Lewis, and this is also going to be presented remotely. The topic is the transformation and destruction of fatty acids by Europa's surface conditions, implications for missions seeking to investigate habitability and life on an ocean world.
Perfect. Thank you, Jamie. If we go to the next slide. Excellent. So when we're looking for life elsewhere in the solar system, one of the mantras we can follow is to follow the water. And if we take that approach, Europa, one of Jupiter's 4 Galilean moons, which I'm showing on the left, Europa is one of the most attractive targets as it likely hosts a vast subsurface liquid water ocean. The crust that overlies that ocean is composed predominantly of water ice, but it also exhibits spectral features indicative of widespread salts. Now, the dynamics of this icy crust may permit the exchange of materials between the ocean and the surface. So that begs the question, could there be molecular evidence of the ocean's habitability and potential life present in the salt deposits that we see on Europa's surface? Next slide, please. As always, there's an issue we have to contend with, and Europa is very much radiation. Any material present at the surface and near surface of Europa is going to be exposed to substantial doses of ionizing radiation due to the moon's orbit being located deep within Jupiter's magnetic field. If you haven't studied radiation before, when I talk about ionizing radiation, that's any radiation that is energetic enough to detach electrons from molecules to form ions. Ionizing radiation consists of subatomic particles or electromagnetic waves such as X-rays and gamma rays, as I'm showing at the bottom. And this presentation will also use the SI unit for absorbed radiation dose, the gray, which is the absorption of 1 joule of energy per kilogram of matter. Next slide, please. So Europa's radiation environment is dominated by electrons and ions that are trapped in Jupiter's magnetic field. And these are constantly bombarding Europa's surface. And they include sulfur and oxygen ions sourced from volcanic eruptions on the innermost Galilean moon Io. So it's cool to think of processes in one world affecting the chemistry of another. And the complexity of the magnetic environment around Europa makes the radiation environment highly spatially variable. And it raises the question, could there be areas that receive slightly lower dose rates compared to others? When these electrons and ions in the magnetic field do impact Europa, they will generate a cascade of secondary products such as gamma rays that can penetrate even deeper into the surface. And our study was interested in how are these harsh radiation conditions going to impact organic compounds relevant to astrobiology. Next slide, please. So for choosing our radiation type and dosage for our laboratory experiments, we went with 1 megaelectron volt gamma rays at doses up to 2 megagray. To put that into context, for most locations on Europa, the upper millimeter of the crust would be subjected to a 10 megagray dose in less than 1,000 years, while materials at depths of around 10 centimeters that are located in those areas of Europa that we suspect might receive those lower doses, a 4 megagray dose is still going to be reached within around 13 million years. So with our 2 megagray dose, we're looking at processes that are going to happen very rapidly on geological timescales on the surface of Europa. Now, at depths beyond a few centimeters to decimeters within Europa's subsurface, those secondary radiation products such as gamma rays will begin to dominate, and that's the kind of depth that Europa Lantern might be sampling in the future. So we wanted to focus on gamma rays for that reason. Additionally, gamma rays interact with matter in a similar way to energetic electrons, so our findings in the lab are relevant to both the subsurface and the immediate surface, and 1 megaelectron volts, gamma rays have the practical advantage of not inducing radioactivity in samples, which makes them much easier to work with. Next slide, please. So the organics we wanted to study were, we decided to utilize fatty acids as they are critical for the synthesis of compounds that form cellular membranes in terrestrial organisms. I'm showing a typical structure of a fatty acid on the top right. And if you look at the caption where I call it a C18-0 fatty acid, that nomenclature, it just means there are 18 carbons in that fatty acid, and the 0 means that there are no double bonds. It's a saturated fatty acid. And fatty acid structures are built from a long hydrocarbon chain, which is hydrophobic, and a carboxylic acid head group, which is hydrophilic. And those two properties mean that fatty acids can spontaneously form vesicles, as I'm showing on the right, that may have acted as protocells in the prebiotic Earth, and potentially in Europa's ocean as well. Fatty acids can also act as long-lived molecular records of ancient life and environmental conditions. And there's also abundant industrial literature that exists for gamma radiation of fatty acids, but they're conducted in much lower doses and at standard temperatures. And in those studies, they typically see that when you gamma irradiate a fatty acid, you tend to cleave between the carboxylic acid group and the long hydrocarbon chain to form what we call a CN-1 alkane. So you knock off one of those carbons and you lose it in the carboxylic acid group. And then if that's replaced by hydrogen, you're left with C17 alkane. That's your CN-1. You can also cleave between the OH, the hydroxyl group, and the rest of the structure. And if that's replaced by hydrogen, you get something called an aldehyde. So these were two molecules we were interested in looking for in our samples. Next slide, please. Now, we have our radiation type, we have our organic. For the salt we wanted to mix our organics with, we based it on the fact that in near-infrared mapping spectrometer data taken from the Galileo spacecraft, there was the widespread presence of hydrated sulfuric acid, and follow-up investigations using the WM Keck Observatory found that there was an unknown infrared absorption feature that correlated with the spatial distribution of sulfuric acid. And that was found to have a spectrum most consistent with a magnesium sulfate brine or a magnesium sulfate heptahydrate. Now, much of that magnesium sulfate could have an origin related to radiation. As we know, there's sulfur and oxygen ions constantly raining down from Io, but also some of it could be sourced from the ocean based on experimental and theoretical models. Next slide, please. So to make our mixture, I took a monohydrated magnesium sulfate powder and mixed it with a dichloromethane solution containing C14 and C24 saturated fatty acids, and then dried that down in a rotary evaporator. And then aliquots of that mixture were then placed into cryogenic ampules that I'm showing on the right. They were flame sealed in the vacuum and then irradiated at up to 0.2, 0.5, 1, and 2 megaray doses at liquid and nitrogen temperatures, which is similar to the global mean temperature that we expect on Europa. The non-irradiated control and the irradiated samples were then examined by a suite of analytical methods. Unfortunately, in the time constraints, I can't get into these individual methods, but I'll include a link to our paper at the end. So if you're interested in the GCMS, EGA, Raman spectroscopy, or LDI work we did with these samples, please take a look at that paper. Next slide. So what did we find? So remember, we're starting with an extremely simple mixture of two saturated fatty acids doped onto a monohydrated magnesium sulfate. And if you look at the column immediately to the right of that, we have indirect evidence that I'll get into in a second that we did form that C14 aldehyde. And we definitely have clear detections of those CN-1 alkanes. We have the C13 alkane, we have the C23 alkane. We had additional detections that weren't reported in those industrial studies. What's interesting is we detected a small population of shorter chain saturated fatty acids. So we're seeing fragmentation that's not occurring around the carboxylic acid group, it's occurring in the hydrocarbon tail of the fatty acids. And that's extremely significant because as astrobiologists, we're interested in the potential signals that could be preserved in fatty acid populations. And here we're seeing radiation forming its own subset of shorter chain saturated fatty acids. Additionally, we saw unsaturation being induced in the starting C14 and C24 fatty acids to form monounsaturated equivalents. And we also saw radiolysis of radiolysis products. So we didn't directly detect the aldehyde. What we did detect was a C27 ketone, which you would form if you joined a C13 alkane fragment with a C14 aldehyde. We also detected a C26 alkane formed by combination of those C13 alkane fragments. So here we're seeing combination occurring to form compounds that are bigger than the fatty acids we started with. We also saw unsaturation being induced in the C23 alkane to form a C23 alkene. And we saw those CN-1 alkanes being formed by radiolysis of those shorter chain saturated fatty acid products. In the interest of time, can we skip 2 slides ahead? Perfect, thank you. So that slide showed that we're introducing a great deal of complexity in terms of the radiolysis products we're forming, but we're also, a key question is also how much of the starting fatty acids are we losing? And when we looked at the data after a 2 megagram dose, we had less than half of the amount of the C14 fatty acid measured compared to the non-irradiated control. So we're seeing substantial losses of those fatty acids in the starting mixture. And that equates to a radiolysis constant that indicates 90% of the starting fatty acid would be destroyed after a 5 megagram dose. And remember, for most locations on Europa, the upper millimeter of the crust will be subjected to a 10 megagram dose in less than 1,000 years. Next slide, please. So to quickly wrap up, there's 2 key points here. Fatty acids associated with near-surface magnesium sulfate deposits on Europa will be rapidly destroyed by radiation, and those radiolysis products will be dominated by CN-1 alkanes, which is consistent with what we see in industry. But under the Europa-like parameters of our investigation, we also observed the formation of unsaturated fatty acids, shorter chain saturated fatty acids, and combined radiolysis products. So when we're searching for these pristine organic signals in irradiated terrains, we're confounded by both substantial losses of those starting signals and the generation of diverse radiolysis products that will scramble any surviving signals from the starting conditions. That QR code will take you to our paper, and please email me or reach out on Blue Sky if you have questions that we don't tackle in the panel. Thank you.
Thank you, James. The third talk in this section will be given by Bernadette Deneen. It's entitled Effects of Chlorine Dioxide Gas on Heterocyclic Aromatic Organic Structures, Implications for Subsurface Biomarker Survivability on Mars.
Hi, everyone. My name is Bernadette. I'm a 3rd-year PhD student at Tufts University, where I work in the Planetary Chemical Analysis and Astrobiology lab. Today, I'm going to be talking about some chloride dioxide gas generation and its effects on potential Martian biomarkers. So I'm sure many of those in this room understand that when we're searching for life on another world or beyond Earth, there are several things that we keep in mind. I've divided those into 3 very simple categories here, and they are where we are looking, how we are looking, and what we are looking for. I will refer back to these categories periodically throughout this presentation. So we know we want to look for life on Mars. There's our where. And we know, given a wealth of prior discovery and research, that Mars is home to both a history and modern presence of environmental factors that can affect the stability of life. These include impact events, solar energy particles, galactic cosmic rays, chemical oxidants, and UV radiation. All of these can individually and cumulatively affect the stability of life as we know it. Today, I will be focusing on the UV irradiation of chemical oxidants on the surface of Mars and how these processes can affect biological compounds or our detection of them. So in 2008, the Phoenix lander detected approximately 0.6 weight % perchlorate, a chemical oxidant, in the Martian soil. And since then, perchlorate has also been found in other areas on Mars, including Martian meteorites, EETA 79001, and Tissant. So the Mars Odyssey orbiter also detected a global chlorine presence on Mars. And prior research in our lab by Carrier and Kunavis in 2015 has shown that the chloride salts can interact with the mineral surfaces and form these oxychlorine compounds on the surface of Mars. But it's likely that these compounds are not sitting inert on the surface of Mars. When we irradiate an oxychlorine like sodium chloride, for example, we can get these generations of chlorine dioxide gas, which we see, which we can see right here. This generation of this gas is a pretty significant issue, given that chlorine dioxide gas is an oxidant. It can oxidize critical cell components. We use it here to purify our water samples. It's a very abrasive gas. And so we have these, interactions here that are currently largely unexplored. So how do we search for life in this environment? How do we search for life as we know it in an environment that can change life as we know it, or degrade life as we know it? Perhaps life is surviving as we don't know it, meaning it is changing its structure in some chemical capacity in order to survive in the Martian environment in a different way than we know it does on Earth. One of the best ways to explore these types of processes is in simulation experiments. And so that is what I do. So I use a Mars simulation chamber where I can constrain the surface of Mars given a variety of habitability factors that include things like low pressures, Mars atmospheric gas mixtures, low humidities or controlled humidities, controlled temperatures, and of course, the Mars solar spectrum. And so it's in these chambers that I can generate chlorine dioxide gas. Working off of work done by Jane et al. In controlled nitrogen inert environments, I irradiate sodium chloride in the Martian environment, dominantly CO2 atmosphere, and I generate chlorine dioxide gas. I can detect this in a variety of different methods. Here we can see the chlorine dioxide anion in my samples using ion chromatography, where it does not exist in my control samples. And so we isolate the effects of this gas using a layered glass filter paper approach, where we put our biological compounds below the filter paper, and we put our surface simulant with the oxychlorine compound or silica base on the filter paper above it. What this means is that we can irradiate the sodium chloride without any of the UV light impacting our biomarker, as there is also plenty of literature that shows that UV light can degrade or alter biomarkers as well. We run these experiments at very short timescales. This gas generates very quickly in under 20 minutes. So we run them at 1, 2, and 3 hour timescales at this time. And so we have the where and the how we're looking for these compounds, but what about what we're looking for? What life are we using? What examples of life are we using? So heterocyclic compounds and macrocyclic compounds are found ubiquitously across life. They are fundamental building blocks to a lot of life essential molecules like DNA, RNA, enzymes, hormones, vitamins. It's in our blood, it's in chlorophyll. And so these structures are really great examples of what we can think of when we look for biomarkers on another planet in terms of life as we know it, particularly porphyrins, where we know that they can be found billions of years old on our own planet. And so for this body of work, we selected an example from each of these categories. So we're using a heterocyclic aromatic amino acid, we're using a nucleoside, and an array of porphyrins to investigate the effects of chlorine dioxide gas on complex structures. And so I'm going to start the results section with an iron porphyrin, both because there's a lot of iron on Mars and because iron porphyrin is also found ubiquitously across nature. And so we monitor these biomarkers in 2 ways. We monitor a primary ion to determine overall degradation of the parent molecule, and we also monitor potential structural changes that these molecules are making in order to survive their environment. So here we can see in this figure right here that at the 1 hour time scale, this iron porphyrin doesn't really show much difference between the control and irradiated sample. It's a very, very small difference. But what we are seeing is that despite this small difference, we're seeing the generation of these new ions seen here in figures A, B, C, and D. And so what this tells me is that even though we're not having a substantial decrease in our porphyrin with this gas exposure, we are still generating new ions. So that could mean that the top layer of this porphyrin is perhaps polymerizing or forming some shield layer that protects the remainder of the parent compound, but is still able to interact with the gas and generate new ions. And so in conjunction with LCMS, we also use a UV-Vis approach to evaluate porphyrins because they have very distinct regions in the UV spectrum, which are the SORAY band, which we see here, and these Q bands, which are zoomed in inlaid here. This is a great method to determine the demethylation of these porphyrins, conjugation changes, or structural substituent changes for porphyrins. And so we can use these methods to compare similar porphyrins, but we have something like hematin here, and then its free base porphyrin, protoporphyrin 9. And so what we're seeing is that with the metalloided porphyrins or the metalloporphyrins is that at the 1 hour time scale, they're losing their metal center, but they're overall retaining their bulk structure. But at the 2 and 3 hour time scales of this chlorine dioxide gas exposure, we're seeing almost a complete degradation in these porphyrin compounds in both our UV-Vis spectra and in LCMS spectra. The caveat to this is at the end of the sample suspension or resuspension, we're seeing this reddish residue that you can see here that is very resistant to all manner of organic solvents that we normally use to dissolve our porphyrins in lab. However, it is very soluble in water, which is interesting because many of our porphyrins that we use exhibit very low solubility in water unless there are substituent changes on the ring or there is a chemical additive. I did not add a chemical additive, and so this would lead me to believe that there are some structural changes taking place with the interaction of this gas. For the analysis of the amino acid tryptophan, we use GC and LCMS analysis. You can see here the main takeaway of the slide is that with an increase in relative humidity, we see a massive drop in tryptophan concentration or recovery. And with the increase in gas exposure time, we're seeing a significant drop in recovery for tryptophan. The last compound I'm going to talk about is the bicyclic purine adenosine. So with adenosine, we aren't really seeing major ion changes, but we are seeing significant changes in abundance, which you can see here at these two scales where we have the control over here and the irradiated sample here, where we have a much lower, approximately a 30% decrease in abundance. What this tells me that even though I'm seeing this decrease in abundance with no new ions, is that there is a potential issue with our post-chamber resuspension, and it's not catching all of the ions that we're generating. And so we would have to work on developing a new method to analyze these. So in conclusion, the main takeaway from this is that we are generating this destructive chlorine dioxide gas in a Martian simulated environment at very low humidity. This gas is penetrating below its generation point, and it is interacting with the biomarkers, the complex biomarkers that we've selected, whether they are altered or they are degrading entirely. The metalloporphyrins are exhibiting some sort of resistance compared to their free base pairs, which is something that we expected given the stability of these metallated porphyrins. And the overall timescales are going to have to be longer to observe major differences. Future work includes different timescales, different gas exposures, temperatures, and expanding the porphyrin array. With that, I would like to thank my PI, Samuel Cunaves, and NASA for funding this work, and you for listening. And I look forward to any questions you guys may have. Thank you.
Thank you, Bernadette. And our final talk in this section will be given by Kristen Twombly. It is quantification of the degradation and fragmentation of adenosine under Martian conditions.
Hi, I'm Kristen, or Molly Twombly. I'm a second-year chemistry PhD student at Tufts University, and today I'm talking about the degradation and fragmentation of adenosine under Martian conditions. So first, everyone in here knows I were looking for life on Mars, but I'm just going to give a quick overview just to put my research into perspective. So this is an image from NASA Goddard Space Flight Center, Big Think, of early Mars and Mars today. And early Mars looks a lot like early Earth and Earth today from satellite images. So both planets had abundant liquid water and carbon dioxide-rich atmosphere and intense volcanic activity, creating the perfect setting for prebiotic chemistry. These shared conditions suggest that if life could arise on Earth, it might also have emerged on Mars. And this is through a process called convergent chemical evolution, where similar environments drive the development of similar biochemical processes. So if Earth had the essential building blocks for life, carbon, water, nitrogen, and phosphates, then Mars also had these essential building blocks for life, carbon, water, nitrogen, and phosphates. And it also had energy-rich environments like hydrothermal systems and redox gradients that could have powered these life-sustaining reactions. There's also minerals like clays and oxides that could have also catalyzed reactions. However, as we know, Mars lost its magnetic field, causing its atmosphere to thin and its surface water to disappear, perhaps halting life processes and causing this divergence in these two planets' fates. Also, I'd like to just add something somewhat tangential before I move on. But by studying Mars, we're not just searching for biosignatures or aliens. We're also trying to prove and determine whether the chemistry of life is a universal consequence of the right conditions, or whether Earth's story is a cosmic norm or an extraordinary circumstance. And so what we're mainly focused on is adenosine as a biomarker. Adenosine is essential to life on Earth because it plays a critical role in genetics and energy transfer. As a nucleoside, adenosine is aromatic and it's comprised of adenine, the nucleobase, and the ribose sugar. It is also a fundamental building block in DNA and RNA. Additionally, adenosine forms ATP, which is adenosine triphosphate, so adenosine with 3 phosphate groups, which is a primary energy carrier in cells. ATP fuels nearly every cellular reaction, and it is in all living things. Animals, plants, and microbes need adenosine because it is integral in cell division, muscle contraction, and nerve signaling in animals, photosynthesis and growth in plants, as well as genetic metabolic processes in microbes. And so moreover, adenosine's role in the genetic code makes it indispensable for replication and transcription processes critical for cell growth. So therefore, when looking for undeniable signs of life, we think of DNA. So adenosine should be a ubiquitous contender for life on Mars. So how are we actually looking for this life on Mars? As Bernadette mentioned, in our lab, we have a Mars simulation chamber, which is shown here with the UV lamp. So in order to get our adenosine into the Mars chamber, we do a series of sample prep, and this is called plating. So we take beakers, and then we have a mixture called SOL, which is sodium, silicon dioxide, ethanol, and water, and that is plated on the bottom of the beaker and left to evaporate, so the ethanol and water can evaporate, leaving behind a solid ground level, which is just the silicon dioxide. Then a 6 ppm mixture of sodium perchlorate and ethanol is then plated on top of that. The ethanol is let to evaporate off, just leaving behind the perchlorate. And then a 10 ppm solution of adenosine and methanol is plated, and the methanol evaporates off, leaving the adenosine. I chose 6 ppm and 10 ppm for my perchlorate and adenosine respectively because it's based off of the limits of detection and the age of our instruments that we have access to in lab. And so then our adenosine will go into the Mars chamber, and we're essentially trying to replicate this depiction from Carrier and Cunavas from 2015. So at the electron level, perchlorate destroys biomarkers through a series of oxidative electron transfer reactions. Perchlorate itself is a highly oxidized form of chlorine, where chlorine is in the +7 oxidation state, meaning it has a strong tendency to accept electrons and be reduced. When heated or irradiated, perchlorate decomposes into reactive intermediates like chlorate, oxygen radicals, and chlorine radicals through homolytic cleavage. These radicals are electron deficient and highly reactive due to their unpaired electrons. So when they encounter these organic molecules, such as adenosine, they want to take their electrons, especially from these electron-rich centers of CH, CC, and CN bonds, which adenosine is full of. So this initiates a chain reaction of bond breaking and fragmenting these organic molecules into their smaller oxidized components. And if that weren't bad enough, perchlorates has a high reduction potential, which makes these electron transfers thermodynamically favorable, which is driving this destruction of the biomarkers. So this electron-driven breakdown explains why detecting intact organics on Mars is so challenging, because any exposed biomarkers are rapidly oxidized and degraded. So if we're not looking for these full biomarkers, and instead we're looking for these broken down signals of adenosine, how are we actually going to look for that? So that brings us to post-chamber analysis. And the first thing we have is a separation. So when our beakers come out of the Mars chamber, we dilute them and then spin them in solution to suspend the solids in their solution. Then we use a Hypercept C18 column to filter out what we don't want. So we condition with methanol and water. Then we apply our sample, which is the plated adenosine, perchlorate, and SOL into the sample. The SOL is eluted because it destroys our instrumentation. We then want to elute any contaminants such as the perchlorate because we're not looking at the oxidized species. We're only looking for adenosine and its fragments. So we use ammonium hydroxide to elute this perchlorate, and then we use a 2% acetic acid solution in order to elute this adenosine. And so once we have adenosine in solution, we're able to do post-chamber analysis, which is the instrumental analysis. So the results that I'm going to be talking about are from a liquid chromatography mass spectrometer, which is an LCMS. And I've included my mobile phase gradient here, which I use water and ACN. And it's about an 11-minute method, so everything is run in triplicate. But in the red box is where I'm looking for adenosine to elute, which you'll see in the subsequent slides. So here are some results of an EIC, which is an extracted ion chromatograph, which is taken at 268.16 m/Z. And adenosine is 267, but since we're running in positive ion mode on our LCMS, we're looking at 268. And so here we can see we have a control sample, which has not been irradiated, but it is plated the exact same way compared to an irradiated 24-hour sample, 48-hour sample, and 72-hour sample. So I'd like to draw your attention to the right-hand side of the graph, which is the axis of abundance. And as we see, as irradiation time is increasing, the concentration or abundance of our parent ion adenosine is decreasing. And now this is a total ion chromatograph. So this is not just the adenosine, but all of the ions in solution compared to a irradiated 24-hour sample, 72-hour sample, and 48-hour sample. I'm going to go through them individually, but very briefly because I'm running out of time. So here we have a 24-hour sample compared to the total, the TIC of the control. And as you can see in the red boxes, we have a formation of new peaks. So here is potential that this could be a new adenosine fragment, or the adenosine is creating a dimer, so it's making these protective groups, so it's becoming harder to break down from the perchlorate. And I'd also like to draw your attention to the concentration on the side, and not only is the concentration of adenosine decreasing, but the total ion concentration is also decreasing. Here's a comparison of the 24-hour TIC to the 48-hour TIC. Again, we see new peaks forming, but as well, we're seeing the peaks that we just saw formed degrade even in a 24-hour increase. As well, the concentration of a 48-hour sample is greatly decreased from a 24-hour sample. And then this is a 24-hour sample TIC and a 72-hour TIC. And then again, we're seeing the orange peaks disappear completely into the yellow, and the concentration is also decreasing. So where does this get us with identification, conclusions, and future work? So the big takeaway is as experimental irradiation time increases, the concentration of adenosine is decreasing. But because our LCMS in our department does not have a database, we need to look elsewhere to start doing ion identification. So as of right now, we're going in by hand and taking all of the new ions that are forming and comparing them to the blanks and the controls, but that's a lot of work. So GCMS methodology is being developed right now, but because adenosine is not volatile, it has a high boiling point, it needs to be derivatized in order to be run through the GCMS. And so the derivatization methods are in development in order to look for these TMS varieties. I know that was a lot of chemistry in a very short amount of time. If you have any questions, you can check us out at planetary.chem.tufts.edu or ask questions. And I'd like to thank my PI, Samuel Kunavas, and NASA for funding these grants.
All right, thank you. And I'm going to turn and invite the panelists back up and turn this over to Eric and Bailey to monitor the in-person discussions. And I'll keep an eye on the virtual Q&A if anyone online wants to ask questions.
I'm going to go all the way over. Can you see that one?
Jonathan Lenine, JPL. For Bernadette, have you looked in your chamber at the organics that have been found on Mars, like thiophene, for example, to see what the effect of these chlorine compounds would be on them? And if so, what is that effect?
Is this on? Okay, great. No, I have not personally looked at those compounds. I'm particularly interested in porphyrin-based compounds. The reason I'm including alternative compounds like polycyclic amino acid or nucleoside in this array is my end goal is to be able to categorize changes based on structural, and so be able to extrapolate that to other similar structures beyond what I am just doing. But I have not done those specific compounds.
Thank you.
Thank you.
Hi, David Bracey from University of Florida. Two questions, one for Amy and one for James. Amy, did you perform other than EGA and TMAH pyrolysis with GCMS after that, and pure methanol extraction to understand whether it was the methanol that helped to extract from the matrix, the compounds, or the TMAH that really break down?
Okay, I think I'm set up here. Thanks, David. And yeah, we can totally talk more about this back at UF when we get home. But so let's see. So the short for the methanol, we actually did collect the methanol and DCM, we attempted to perform a Bly Dyer extraction on it to see what the organics were that we had like leached off of presumably the surface and any cracks in the rock. And unfortunately, it did not work. So the good news, I guess, is we have plenty of samples sitting in bags from Dorothy's garage. And so hopefully we can revisit that and try again. And then we have pyrolysis, like just neat pyrolysis for a handful of samples, but not that full suite that I showed. But that's actually something that I want to revisit now that we've kind of seen how the FAMEs and the alkanes are playing in the samples from the TMAH thermochemolysis experiments.
Great, thanks. And for James, so you quickly showed the rates of the kinetics for the C14, but not for the C22. Doing exactly the same experiments for Titan, but with water or water plus ammonia, we saw that for short fatty acids, there were a linear degradation, but for high molecular weight, it was either exponential or polynomial kinetics degradation. Did you see a specific pattern, C22 versus C14?
Thank you for asking that question, because if I had more time to speak, that's something I would have gotten into. Because we deposited our fatty acids onto the magnesium sulfate from solution, we found that the C14 was deposited homogeneously across the powder, whereas the C24, because it's such a big structure, that came out of solution very quickly and it formed these clusters. So we were seeing a great deal of heterogeneity. So we didn't feel confident enough to calculate a radialis constant for the C24. So we just looked at the C14. So it would be really interesting, as you say, to look at these larger structures in future studies to see how they're behaving.
Great, thanks.
Jeff Osterhout, Caltech. My question's for Amy. Amy, you showed some plots with some TOC values in there, and I was just curious if there was any relationship between the TOC and the organic yields that you got. And also if you looked at any different metrics like hydrogen to carbon ratios to get a general idea of thermal maturity.
Thanks for the question. So I'll preface this by saying that this all started out as sort of an undergraduate research project. And so I've sort of taken it over from two undergrads who did a great job kind of starting out on this. So I have not done C to H. I think that I would be very interested in looking at like the degree of aromatization in the samples, maybe using NMR. And then what was the other question?
The TOC values.
Oh, yeah. So I did look at I did not see the correlation. I found that really interesting. Some of the samples that had kind of the best alkane yield and fatty acids as well did not come out as sort of the highest TOC values. So there's a lot to still dig into with this project.
Yeah, interesting. Thanks.
Hi, Bonnie Chase, JPL. My question's also for Amy. Amy, it's so nice to see you virtually. When you were presenting, I was really struck by some parallels with the sample history and what you were finding with some of Jock and Brock's work in the Archean on hopanes and steranes. And I think they had gotten around that by doing exterior versus interior comparisons and slice experiments. I wondered if you could talk some more about the sizes of your samples, the permeability maybe of the chert, and whether you have any plans to try to get some more pristine sample.
All right, great questions. And Bonnie, at some point in our lives, we will actually be in the same place at the same time. I don't think it's happened yet. So getting new samples, I think, is not an option right now, and especially collecting them the way that we would prefer to, like trying to reduce any contamination as much as possible. But let's see here. Dang, I keep forgetting these things. What was the first question?
So my question was about the size of the samples and whether you considered trying to get like to the middle.
Interior versus exterior. Yes. Yeah. So we did start this project several years ago now. So I'm trying to recall like how big the chunks were, but we had like pretty big chunks that came in the bags, right? And so then we were trying to crack that open and get to an interior sample. We did do a complementary exterior, but I mean, I think like at least that would be relatively easy to do because we still do have those samples. I don't remember the porosity of the cherts, but that is definitely something I'd be happy to follow up with you on moving forward, because I'd be curious about your porosity insights and thinking about how we might be liberating organics or not from these samples.
Thanks, Amy. Hi, this is Aditi from LPI and JSC. I have a question for the chlorate perchlorate oxidation group. So I was wondering if you have thought about or looked into any sort of mineral organic interactions that might increase the resistance of these biomarkers that you're studying to the oxidation by the irradiated chloride or perchlorate species.
Yeah, so the mineral composition is a really interesting thing. So there's a bunch of labs out there that are investigating the protective effects of different minerals, which is something that we do want to explore as a next step. We don't have specific minerals yet. We're thinking of labradorite, but we're definitely looking into these types of minerals. It's just like it's so far away because with how these experiments are run, we have to introduce each variable very like not slowly, but individually, and then do a bunch of experiments on that one variable. And then if we have multiple variables, we have to do different combinations, right? We have to combine 1 and 3, 1 and 2 before we do 1, 2, and 3. So it's going to be a while before we get there.
But if you're interested, I'd be happy to collaborate on that.
Yes, Aditi, reach out to me. Let's collaborate.
Hi, this is Xiaowei Wang from Cornell University, and I have a question for Dini. It's like for your experiments. You probably these are experimental details, but I'm sorry that you probably mentioned, but I didn't catch that. So it's like in your experiments, is your organic dissolved in water or just like solid phase?
Thank you for your question. No, so we start with stock solutions of our compounds to make sure that we're trying to get an exact concentration in our dishes, but we do evaporate them before putting them into the chamber. We use a reactotherm, so we evaporate them under nitrogen.
Okay. Oh, so it's like basically just like also it's like already evaporate. So it's mostly solid phase.
Yes.
Okay, great. Thank you.
Thank you.
There's a question online from Maria Cortez. I believe it is for Kristen. How does increasing experimental irradiation time affect the molecular stability of adenosine? And what implications does this have for detecting nucleobases in extraterrestrial environments?
Thank you. That was a great question. So what we've found right now is that as increasing the time, the perchlorate and the SOL and the irradiation are kind of oxidating the adenosine and breaking it down and fragmenting. I couldn't really get into it because I didn't have enough time, but we do have lists of potential fragments, so we have those options. For other nucleobases, right now I'm just kind of tackling adenine, adenosine, and ATP, but there has been work done in our lab on alanine before. Did that answer the question?
I think so. It was a written question, so we'll see if that person has any follow-up. But I don't see anyone at the microphone, so I might use session chair's prerogative here and ask a couple questions. I have one for James, and Maria just responded that yes, that did answer her question. I have a question for James and then one for Bernadette and Kristen together. So James, it looks like it gets very complicated. Like a single fatty acid can produce a really complex mixture of products after your irradiation, right? So that's going to very much complicate looking for biosignatures. But I wonder if you took a complex mixture of fatty acids that showed like an even over odd preference, like you might assume would be a biosignature, and you irradiated that, would you see any kind of similar bias in like odd alkanes being produced, or anything that might still stand out as a pattern of biological origin? Or do you think it would all just be too complex to be able to pick any of that out?
Thank you, Jamie. That's a fantastic question because I can touch on two things that I can touch on in the time allowed. And the first is I worry like my talk comes across as being a bit of a Debbie Downer when it comes to organic chemistry in the solar system. But we have to remember is one, we're looking for organic signals, but also we want to understand what organics could potentially be transported from Europa's surface down to the ocean. So what makes our exploration difficult could actually be a important source of organic resources for life in Europa's ocean. So I think my answer to your question is that I would worry that we could, as soon as we get into irradiated materials, I would worry about trusting the interpretation we're seeing, because we're not just seeing a starting signal and then a slow loss of that signal. We're seeing those products being transformed into a huge array of different radiolysis products. And we're also seeing the combination of some of those. So yes, we might be getting those alkanes showing that kind of initial even over odd predominance in the fatty acids. But then what if some of those alkanes join together? And then what if those alkanes then undergo radiolysis of their own? It gets very difficult to interpret, but we have to remember whatever we detect on the surface of Europa eventually, that's going to be very informative. It'll be very challenging to see biosignatures in anything that's been heavily irradiated, but it's going to be very informative to know, all right, this organic assemblage that seems to be stable in this highly irradiated terrain, what would happen if that gets fed down into the ocean? What kind of life might that support?
Great, that's really cool. Thank you. And since I still don't think I see anyone at the microphones, I'll ask a question for the last two speakers. I really enjoyed seeing those talks back to back because you're each, I know you're in the same lab, but you're focusing on different areas, the effects of the chlorine dioxide gas or the radiation in the solid state. How do you think the two of you might work together, like using your results and combine to understand more about the overall, what might be detected on Mars? I see you've probably thought about this.
Yeah, we've actually thought about writing a paper together. There's a horror movie called As Above, So Below. And we were going to talk about adenosine and biomolecules as above, like on the surface, and below with permeation. It's a work in progress.
That's excellent.
We still got a few minutes here. I'd like to ask a question if I can. I have a question for Bernadette. You were analyzing tryptophan as in terms of how it reacted with ClO2. Did you get a chance to look to see if there was any difference in how tryptophan reacted with ClO2 based on chirality?
I did not. I only use L-tryptophan in my work. We don't really focus on chirality in our lab. I think it's really interesting. It's just typically beyond the scope of our regular analysis. Great question.
Your analysis for tryptophan, I think if I remember correctly, you said you use both GCMS and LCMS.
Yes.
Now, if you focused on the L enantiomer exclusively, is that a analytical limitation? Meaning let me clarify. Meaning maybe there's some inability to distinguish analytically between the two enantiomers, or is it just what reagents are available in the lab?
It's really just what reagents are available in the lab. We're mostly focusing on, I'm mostly interested in like ring breaking. And so while the enantiomeric aspect of tryptophan it's really cool, it's just not something that I focus on. The reason I do LC and GCMS analysis is to kind of follow different ions through my sample analysis, because there are some things that are going to work much better with a volatile based instrument and some things that are going to work much better in an aqueous based instrument.
All right, I'll ask another question for Kristen. You showed a nice plot of nice results about how increasing irradiation time led to a reduction in adenosine concentration. Do you have recommendations of a concentration reduction rate, or what types of concentrations you're seeing being depleted as irradiation time increases?
Yeah, we haven't really looked into that yet. We're mostly just trying to see if it even will be decreased and see if it is fragmenting with the irradiation in the perchlorate, but that's definitely something to look into to see how much of a decrease we're seeing from the control to each irradiated sample.
So at this point, it's not possible to give a relative loss rate or a semi-quantitative loss rate at this point, right?
Not to my knowledge, not necessarily.
On the topic of loss rates, did you see any loss of adenosine during sample prep through the multi-step process that was used?
Like my SPE separation. Yeah, so there could be adenosine coming out, but because adenosine is so polar, the solutions that I'm using shouldn't allow adenosine to be eluting, but they could elute other fragments of adenosine that aren't as polar. So there could be like sample loss for fragments that happened in the Mars chamber, but I wouldn't necessarily know how to eliminate all of that by still being able to eliminate the sol through the micron filter and the perchlorate with the ammonium acetate.
So would it be safe to say that you would expect a minimal amount of loss of adenosine or its fragments during sample prep?
Yes.
Okay, great.
Amy, I see you wrote in the chat, do you have a question you want to ask? Go ahead.
Yeah, no, I can direct the question at the two in-person speakers as well. So yeah, I was also like really struck with how like the complementary nature of your work, like how it can come together. And I was curious, I have a different experience with a Mars chamber, and so I just wanted to ask, so you have your radiation source, you have the perchlorates, you said you're talking about your using different mineral matrices at some point in the future. Can you remind me, in case you said it, I'm sorry, I missed it, but what is do you use like a Mars atmospheric mix in the chamber? And yeah, okay, I see nodding. And then what temperature do you hold the chamber at, and how do you maintain that? I'm just kind of curious about like how different Mars chambers and how conditions, change.
So we use a Mars gas mixture that we order from Airgas. And so it's dominantly CO2 with some small amounts of nitrogen, argon. And so that's how we pump it in. And then as for your temperature question, so we have a cold plate. It's a 25 centimeter diameter in our Mars chamber that we can bring down to -80 centigrade. And so we currently are running our experiments at 25 C and 4C. We've not gone negative yet, but we do intend to.
All right,
It's
that was
just a
my
matter of
follow-up
like,
question.
yeah, it's just running that cold plate is actually, to be honest, kind of annoying. And so if we can get all of our control data at a temperature that we don't need to run it, that would be ideal.
That's my experience as well, having the LN2 to get your cold plate where you want it. If you want to work at the really low temperatures, it's a huge pain. So yeah, awesome. I'll have to follow up with you all because I'm curious about more of your setup, but that's fabulous. Thank you.
So that does conclude the Q&A. If we can give our presenters and everyone who has a question one more round of applause. And I'll send it to James to introduce the next speaker.
So the next speaker will be Denise Buchner, and the title is Machine Learning Analysis of Mass Spectrometry Data Can Differentiate Organic Distributions in Meteoritic and Terrestrial Geologic Samples.
Thank you. I hope you guys can hear me. So thanks for the introduction. So you can go to the next slide. In the search for life beyond Earth, the ability to discern whether organic molecules formed through biotic or abiotic processes is really important. And because synthesis mechanisms differ, biotic and abiotic organics display differences in their individual structures and their overall distributions within natural samples. So biotic organics, like those found in samples from Earth and illustrated in the blue box, display structures that are dictated by biological necessity. For example, the hopane or the isoprenoid, which are both cell membrane components. Abiotic organics, on the other hand, like those found in carbonaceous meteorites and shown in the red box, form through astrochemical reactions of opportunity. And while many of the same classes of organics are produced, the individual configurations of those molecules and their distributions in samples are different. For example, the polycyclic aromatic hydrocarbon shown with the 4 rings. Understanding how the molecules different from these two pools is important for interpreting the origin of organics found on Mars and other planets beyond Earth. Over recent decades, there's been a lot of research into biomarkers and meteoritic organics and using these compounds as molecular fossils in the search for life. And while differences in individual molecules and isolated classes of organics have been compared across those two sample types, the full inventory of soluble organics has not been systematically assessed. And this is important when you consider the molecules that fall in that middle box and are known to form through both biological and non-biological processes, and should be expected to exist alongside biomarkers in these complex mixtures of organics found in natural samples. Next slide. So our work takes the approach of looking at this whole profile of soluble organics extracted from natural samples, and we're using machine learning approaches to discover and deconvolve differences in mass spectrometry data, which is important preparation for things like Mars sample return. Natural samples can contain thousands of individual compounds, and approaches like untargeted two-dimensional gas chromatography, high-resolution time-of-flight mass spectrometry can separate and resolve these complex mixtures. At the bottom of the screen, you can see two chromatograms of soluble organics extracted from the Murchison meteorite on the top and the Green River shale from Earth on the bottom, produced by that 2D GCG-CMS. The x-axis is retention time in the first dimension, and the y-axis is retention time in the second, which is a way to separate out the compounds based on their mass-to-charge ratio, or m/z value. Each colored spot represents a compound, and to identify what those compounds are, imagine the chromatogram extends in the third dimension, where the z-axis is that m/z value, representing either an organic molecule or a fragment ion of that molecule produced during the analysis process, and all that information is contained within the colored peaks. Ingesting and analyzing this large amount of information between meteoritic and terrestrial samples is pretty impossible for a human to do, but machines can help us by quantifying differences in these multidimensional data sets to identify patterns in overall profiles and learn differences. And this can form an astrobiological network or framework for analyzing samples beyond Earth. Next slide. So a brief overview of the study. First, we extracted soluble organics from a collection of terrestrial and meteoritic samples and analyzed them with GCG-CMS. Second, we curated that mass spectrometry data into an ML-readable format. And third, we used the data to develop LifeTracer, our biotic-abiotic classifier that can predict the origin of unseen samples from the two pools with greater than 90% accuracy. Next slide. And next slide again. Going into a little bit more detail, first is the experimental portion, where we selected 10 samples from analog environments on Earth and 8 from meteorites, performed wet chemistry extraction to liberate the soluble organics of nonpolar and semipolar fractions, and analyzed them with GCG-CMS, producing that 2D chromatogram you see in blue, which is a representation of a 3D chromatogram when you consider that m/z data extends in the third dimension, but is stacked together into those individual peaks. Next slide. So next we had to convert the blue chromatograms into an ML-readable format, illustrated in the black chromatograms on this slide. In the top panel, A, you can see how that m/z data comes into play. So we took the GCG-CMS data and created numerous chromatograms that we're calling total ion images, or TIIs, from each of our 18 samples, which is illustrated as A for 3 of our samples. For the TIIs, the X and Y axes are still the retention times in the first and second dimension, but we separated out each of the peaks into slices defined by the individual m/z values from 30 to 700 AMU for each sample. So you can see them stacked in the Z direction as individual images, and in the end, that resulted in a total of 12,060 TIIs across the 18 samples. So next we took all of those individual TIIs from the different samples and compared them. In B, you can see a combination of 3 TIIs, each with the same m/z value of 128, and that's from the Green River shale, the Aguasarcas meteorite, and the Murchison meteorite. So the red and blue dots show where in the retention time space those fragment ions are found. The fragment ions can represent either a parent compound or a fragment of the compound, so when a peak occupies a different retention time, it indicates a different compound. Those features were then mapped into this binary matrix, and in total we found 9,152 features that are defined by the triplet m/z, retention time 1, retention time 2, across all the TIIs, which were detected in 1 or more samples, and that was used to train the algorithm. Next slide. So LifeTracer is our biotic-abiotic classifier, and we used a logistic regression model with L2 norm regularization through an exercise of 9-fold cross-validation with nested leave one out cross-validation across the 18 samples. Basically what that means is we just gave LifeTracer 16 of our 18 total samples and told it whether those samples were terrestrial or meteoritic. Then after learning that data, we fed in the 2 remaining unseen samples and asked it to make a classification, if it's more similar to the biotic or more similar to the abiotic, and repeated that exercise until all possible pairs were studied. After the exercise was complete, the trained life tracer algorithm that we produced demonstrates the ability to correctly identify whether those two unseen samples were from the biotic or abiotic sets with more than 90% accuracy. And the figure over here shows the total integrated data set from all 18 samples with retention time 1, retention time 2, and M/Z on the 3 different axes. The red dots are features most closely associated with abiotic samples, and the blue ones are more indicative of biotic. And larger spheres are the features with greater regression coefficients or more important for classification in one direction or the other. Next slide. And next slide again. So we have 3 major results from the study. The first is that the overall profiles of the non and semipolar soluble organics are different for the biotic and abiotic samples. This is pretty much to be expected. So the figure on the right shows a 3D map of features for all of the abiotic samples on the top and all of the biotic samples on the bottom, and you can see that the distributions tend to differ. The bar graphs on the bottom show how they differ with the frequency distributions of those features by M/Z in A, retention time 1 in B, and retention time 2 in C, and red is abiotic and blue is biotic. So the distributions of the M/Z values and the retention times in the first dimension are statistically distinct for our 2 sample types, but not for the retention time in the second dimension. And meteorites tend to have smaller M/Z values and lower retention time ones, which corresponds to a greater number of small volatile compounds compared to the samples that have input from biology. Next slide. The second result is that machine learning of GC-GCMS data can identify individual organic compounds that are shared across samples, and those compounds can be used to help predict origin. From all of those 9,000 plus fragment ion features, we found 140 individual molecules that are shared across 2 or more samples. Determining the identity of those individual molecules requires manual analysis, so LifeTracer can identify features of interest, but it can't reconstruct molecular signatures without the help of a human. An important thing is that some of these 140 compounds are only found in biotic or abiotic samples, but others are shared and found in samples of both origin. So that figure on the right from the previous slide again shows how the features from all 18 samples plot. And from that data, we found that polycyclic aromatic hydrocarbons are top predictive features from those non and semipolar organic extracts, and naphthalene and 2-methylnaphthalene illustrated are the ones that most commonly appear in meteorite samples, and hexamethylnaphthalene was the top predictive compound for biotic samples. Next slide. So the third and final result is that our LifeTracer algorithm can correctly classify unseen samples as biotic or abiotic with greater than 90% accuracy using M/Z values, ion abundances, and the retention time data. And this is an important outcome because with our ML forward approach, identification of individual diagnostic molecules isn't a requirement for being able to differentiate between biotic and abiotic, but instead it's differences in the overall organic profiles. And a human can't really compute that without a machine's help, but this demonstrates that a rich amount of information is contained within organic extracts. So even when many of the same molecules appear in both biotic and abiotic samples, nuances in how the molecules are distributed can give a hint of where they came from. So it means that when you're examining organic extracts from a sample beyond Earth, you can look for individual biomarkers and algorithmically assess the whole sample, looking for a profile-based biosignature. And LifeTracer is a powerful tool that could be used to analyze samples brought back by Mars sample return and look for patterns that could be consistent with life or its absence. And next slide. Here's just the summary again. I won't belabor the point, but I'd like to thank all of my collaborators at NASA, Georgia Tech, and ORAU, in particular the NASA NPP program. Thank you.
Okay, the next presentation should be from Joey Postersky, and it's titled Laser Desorption Mass Spectrometry of Multiple Titan CHN compound analogs with and without prebiotic compounds.
Great. First off, I just want to thank the both virtual and in-person hosts for having me here. I also want to take a second to thank the huge amount of collaborators that's on this project. We really couldn't do it without them, and everything that I'm going to show here is based off of everyone's work and decades of previous analysis. So I just want to shout that out first. And I want to pause here for just a second and introduce Titan, because I realized when I was making these slides, I'm very used to people being understanding of Titan and knowing how alien it really is. And then I was talking to someone and realized that I hadn't really introduced what these different dunes and features are on Titan. So I'm going to take a second and set the stage for what Dragonfly actually is going to be analyzing. So on Titan, we have these dunes that are hundreds of meters tall. And on Earth, we think these dunes are made of sand. On Earth, we know they're made of sand. But on Titan, we think they're actually made of organic compounds that sort of fall out of the atmosphere from an organic haze. So we have these giant dunes made of organic compounds. We also have a bedrock that is composed primarily of water ice with other secondary compounds like ammonia in them. So we have this water ice bedrock, giant sand dunes of organics, and laboratory studies of analogs of these types of organics mixed with water have shown that you can actually get nucleobases and amino acids out of those different mixtures. So we think that Titan may have this pretty diverse and rich prebiotic inventory. So one of the main goals of Dragonfly is to go to Titan to see what is that prebiotic inventory, and if there is life there, be able to detect it. But that is not the main goal, just as a statement. All right. So again, on Titan, Dragonfly will likely encounter these two large classes of prebiotic compounds. So CHN compounds, which form the organic dunes, which is where we're going to land in the beginning, as well as prebiotic compounds such as nucleobases and amino acids, which would be forming from a mixture of the prebiotic compounds, the CHN compounds, with liquid water at the Selk impact structure. So we're going to land in the dunes and then fly to the Selk impact structure, sampling along the way. The Dragonfly mass spectrometer will be the primary means of assessing the organic inventory of samples analyzed on Titan. And here I'll be showing data using the Dragonfly mass spectrometry laser desorption mass spectrometry breadboard at Goddard, including analysis of CHN compounds, so laboratory analogs of what we think is on Titan, prebiotic compounds relevant to Titan, as well as mixtures of both, highlighting what we can actually do on Titan, or what we might be able to do on Titan. So the two broad types of analysis or modes of analysis that the DRAMS instrument will actually use are laser desorption mass spectrometry and paralysis GCMS mass spectrometry, really paralysis stereotization GCMS mass spectrometry. LDMS will be used as the primary mode of analysis. So essentially, it's a broad survey mode where you can scoop up sample, shoot it with the laser, and see what kind of ions you produce. So there's no sample preparation that you need. But what that also means is all ions that you have, both prebiotic compounds and potential CHN compounds, will be mixed in that spectra. If in the LDMS spectra, things are interesting or worth doing further analysis for, we'll then pass that same sample or a similar sample to the paralysis stereotization GCMS instrument to do more to gather more information on chemical structure and chirality and enantiomeric excess, et cetera. Because of the time constraints of a 10-minute talk, today I'm not going to be talking about the GCMS data, although we do have GCMS data for this for these solens I'll be showing here, but just focusing on the LDMS data for today. So this is the Dragonfly mass spectrometer LDMS breadboard that I get to use at Goddard. It's extremely fun. It's a very sciencey type of instrument where you have to flip a bunch of switches. I get to use cryo mode where there's LN2 spewing out everywhere, and it feels very intense. It's a great time. But some of the really important things about the DRAMS LDMS breadboard is that it, as closely as possible, simulates what LDMS is going to be like on Titan. So we actually have two parts of this instrument here. You have the bottom portion, which is labeled LD chamber on the slide. That's where we actually do the laser desorption and ionization. And we do that at DRAMS relevant conditions. So that's going to be at 95% nitrogen, 5% methane at 25 torr, which is what will be happening on Titan, using a 266 nanometer laser, which will be the same wavelength that we're using on Titan. And all of those ions are directed into the ion trap mass spectrometer, or ITMS, which is the same configuration, same geometry as the ion trap mass spectrometer will be actually operating on Titan. So this is as close to you as possible, as close as you can get to operating on Titan. But we have an engineering model as well, and I am not allowed to put tholins in the engineering model. So this is where tholins are going to go probably until the mission's actually flying. And the main goals of what I'll be showing here are really to produce fingerprints of these complex CHN compounds, to compare spectra from multiple different types of complex CHN compounds, and then to determine if we can actually see the spectra from those prebiotic compounds, such as nucleobases and amino acids, when I mix those compounds with the complex CHN compounds. So here are what the actual samples look like. On the left side of the screen are the tholins. So tholins is the term for laboratory analogs of complex CHN compounds that are on Titan. So tholins aren't on Titan, but tholins are on Earth, but they do represent the chemistries that we think are present on Titan. Tholins are made in different labs across the world. I have right now 3 samples in my lab. Here I'll be showing results from 2 different analyses, one from Johns Hopkins University from Sarah Hurst's lab from the phaser tholin set, and another is Cyril Zopa and Thomas Gauthier at Latmos in France. They have another tholin set. I actually have a 3rd set from Xingting Yu and collaborators at University of Texas San Antonio. I don't have that data here, but I'm also accepting tholins from anyone who wants to give them to me for this analysis. I need about 1 milligram, and I can do probably 1,000 different analyses on them. So send them my way, and I will do the analysis. We also have a lot of different prebiotic compounds. I've previously analyzed all of these prebiotic compounds in the LDMS system, so I know what the spectra looks like in our Barry Drams-like analysis. So we have spectra from all these compounds, and we're going to mix some of these with these tholins to see what that spectra actually looks like. So the way I figured I'm going to set this up is we're just going to be on Titan doing some analysis. So right now we're in location 1 on Titan. We made it. What an incredible time. We've landed in our spot in the dunes. We sampled our first tholin. We did an LDMS analysis, and this is what we get. That's so easy. Can you guys figure it out? No, right? Yeah. So this is what tholin spectra looks like. So again, these are very complex CHN compounds that form from semi-chaotic reactions that happen in the upper atmosphere most likely and rain down to the surface. So the structures are pretty wild. We can start to zoom in, maybe start to label some peaks. That's very helpful. So we can compare one group of tholins to another group of tholins. And these are the phaser tholins, our location 1. One ion I'm going to point out here is 144, which is consistent with naphthylamine, which actually, if it ties into the last talk, is abiotic. So we do have an abiotic naphthalene in our sample as well. And from here, we can actually zoom in and do a different mass spectrometry mode and do a high resolution mode. And we can get higher resolution spectra, which is very helpful for starting to make this fingerprint. So now we have this fingerprint of these tholins, which we can start to use to actually identify these tholins relative to other tholin sets. So this is our second tholin set. These are the POMP tholins. One thing I want to point out that's really important is we have this lack of the naphthylamine peak relative to the phaser tholins. So we actually found that this peak, this methylnaphthylamine, is something that is in phaser tholins but not in POMP tholins and is absolutely recognizable in our LDMS spectra. So this is showing that we can actually distinguish different types of tholins in our LDMS spectra. And to point out that these 2 different types of tholins, while made across the world, were both made with 95% nitrogen, 5% methane, using cold plasma ionization sources. So these tholins are made in very similar conditions and still have these somewhat distinguishing peaks, while also noting that there are different aspects of the spectra that are very similar. So we have a lot of similarity and some differences that are detectable via LDMS. Perfect. To zoom in and look at these in high mass resolution mode, we can see that, again, naphthylamine is missing from this top spectra, and there's a lot of other data we can go into. But for now, we're going to zoom on to the next part, saying that we can, in fact, use LDMS to distinguish these different types of tholins. During mission operations, if we saw differences in spectra representative of different types of compositions, we could actually then make that distinction of passing that sample on to the pyrolysis derivatization GCMS to do structural analysis and determine if there's any compounds that are prebiotically or biotically interesting in those samples. But are Titan-relevant prebiotic compounds also detectable when you mix them with these complex CHN compounds? Yes. This is adenine mixed with tholins, ribose mixed with tholins. Just to prove that these actually are mixed with tholins, I want to note that the tholin spectra is still there, but the primary peaks are these different prebiotic compounds. And this also holds for myristic acids, so fatty acids, as well as aspartic acid. So just to wrap up, trams like LDMS and pyrolysis derivatization GCMS will be useful for distinguishing CHN compounds on Titan. Prebiotic compounds can be detectable even when mixed with these complex CHN compounds. And so we will be able to distinguish these different types of compounds on Titan. All right, thanks.
Okay, the next talk is from George Cooper, and it's titled Deuterated Molecules on Interstellar Grains, a role in the formation of early enantiomer excesses in organic compounds?
Okay, thanks. Forward back. Okay, so we want to look at the role of deuterium as possibly creating enantiomer excesses. And of course, there are many forces to create organics on grains, including radiation and magnetic fields. So today, that the laser. No. Yeah. So today I'll be mostly concerned with sugar derivatives, specifically sugar acids. So just mentally add another oxygen to each sugar and you get the acid. So instead of erythrose, erythronic acid, 3 carbon, 4 carbon, 5 carbon. That's what we'll be discussing. And by now, everyone knows why mirror images are important. Enantiomers, for example, why is DNA and RNA and proteins only one mirror image of each organic or enantiomer pair? So did interstellar processes have anything to do with this? Can we do it in the lab? In the meteorites, there are D excesses in the sugar derivatives. So can we reproduce that? And I have a reference later, but we've talked about this a little bit before. What we do, take a sample, expose it to concurrent magnetic and photo fields, pretty mild radiation. That's important. The two together are chiral force, not one alone. And let's see if we can get enantiomer excesses. We analyze by GCMS after derivatization. And here, we presented some of this before, so I won't take long on this. So before I get to the new stuff, I'll show this one again. Previous experiment, we used our forces, radiation, magnetism, one magnetic direction. We got, say, an L of erythronic, one of the 4 carbon compounds, over to D, change magnetic directions. And sure enough, this is what you want to see. Did you reverse the enantiomer excess, which means you can make it prebiotically, possibly. The same thing for its twin 4 carbon compound, threonic. Big D excess and reverse. Previously, even last year, I said, well, look at that big, ridiculous excess of D. Could that be contamination? And I knew, and probably biologists in the audience knew, that if this is contamination, where in the world is the D erythrose and erythronic? This, when you accidentally get something in your test tube or fungus, contamination, erythronic should be through the roof, and arabinose also. So why are these two smaller? If this is a contaminant, they should be much larger. But we would have published this already, except for the fact that inconsistent. Getting any excess, enantiomer excess, you might have to do 5, 6, 10 experiments. When you do get an excess, it is reversible, so that's good. But inconsistencies. So keep doing experiments. Do another isotope label for the sake of contamination checks. Here's one we did with deuterated paraformaldehyde. What did we get? Nothing. Perfectly racemic. You don't want to see that. Okay. And again, here are the structures just for, scale here. So we've done this, we did it 4, 5, 6 times, racemic. And it's like, okay, well, come on. Now, we did notice that the smallest, that's usually racemic in meteorites and most lab experiments, did have what looks like a little bit of AL excess. This is glyceric, the 3 carbon oxidized version of glyceraldehyde. But chromatography, people will know that, well, just because the L is taller, it could be, as my intern said in the past, yeah, the L is taller, but the D is fatter. Right? So look at the area, they might be about the same, so no big deal. The D, I showed the most extreme example. The D, even here, this may fool you. The D may not be much of an excess at all. Glyceric and erythronic are really finicky. Sometimes you can separate them, sometimes you can't. So I'll concentrate later more on threonic. So the D, most of the time, is either actually just like this, pretty much racemic, or maybe there's a little bit more D. But then you notice that it's never like this. 4, 5, 6 experiments, even though this is murky, there's a little bit more, the D is never like that. It's always like this. So in a moment of logic that Sherlock Holmes would have been proud of, we said, well, maybe something is happening here, who knows? Okay, oops, how did that? Skip the slide here. Okay, so we should go back here to a few slides ago, and most people have seen this. You have D and L. The most common example of an enantiomer is carbon with 4 different groups from your textbooks, just differing by their orientation in space. Why is that important? Well, the famous experiment by Bonner showed 2 mirror images, 2 enantiomers, and he shined a chiral force, an asymmetric force, right or left circularly polarized light on the mixture. And sure enough, both absorb the UV light, both are degraded, but one is degraded a little bit more than the other. One, almost 2 here. Reverse the magnetic direction, use left circularly polarized light, right and left, switch it up, and you have an excess of a little bit of the other enantiomer. So it's just a quick example of why you want chiral molecules anywhere prebiotically. Okay, so ignore this word hydrate here. We wanted to, for this, we decided to forget new results. We put the newest results. So I scrapped the whole presentation, and you will see a plain Jane slide at the end. But anyway, moving on. So making sugars, this is how you do it. Put formaldehyde, or prebiotically, and in the lab today, put formaldehyde in water. But what you have in water is not that. You have 99.9% dehydrated methylene glycol or digemdiol, not this. And it's not chiral, even though now you have carbon with 4 different groups. It's not chiral, okay? But on interstellar grains, you could have, I have one paper, and I have to do more literature searching, but one paper where the combined deuterated paraformaldehyde is just as much as the plain old hydrogenated. Plus, there's a lot of water, deuterated water. So now you don't have that when you put this in water. You should have this that on paper looks chiral, and it is, but you're talking 40, 50 Kelvin. So that is now chiral, okay? Also, radiation from here, you make the 2 carbon, that's no news. Now you have chiral glycoaldehyde. Forget that D, it doesn't matter if this is D or H, this is now chiral. Okay. And just a reminder, these 2, formaldehyde, glycoaldehyde, cascade their properties into the larger sugars. So what happens, long story short, what happens when you, what happens when you do this in the lab, do the experiment, put this, what happens? Oops. Okay. Oh, come on here. This is okay, well, it got back there. I don't know how that happened. But anyway, this is what happens. You get an L excess in one direction, a D in another. Same type of sample, you get an L excess. Here, we didn't have time to put this one on. This one is racemic, as I showed you previously. So these had deuterated paraformaldehyde and deuterated water, whereas before we got racemic. So that's basically it. That's the very last thing we got. But this looks repeatable. We did these within hours of each other. So instead of the chromatogram I showed you before that has nice reversals, but it's inconsistent every 10 experiments, we did these back to back to back. I have this one, the racemic one, on my computer. Anyone can see that. So I'll skip the okay.
Apologies for jumping
Well,
in. We need to move to the final speaker soon.
Okay. So deuterium and deuterated water did the trick.
Perfect. Thank you. Okay, the final speaker is Shelby Osborne-Young, and the title is Molecular Dynamic Simulations of Protein Structural Change Induced by KO and Cosmotropic Brine Solutions.
Hello, everybody. Thank you for being here with me today. I'm very excited to show you some cool results that I've uncovered while doing virtual molecular dynamics. What I've done is I've taken an unexposed ancient protein with very simple structure, and I've exposed it to various brine solutions that are found on the Hofmeister series to see how these brine solutions affect the structure of my ancient protein. What just happened? Okay, I got it. Okay, so real quick, I'm going to discuss with you guys about what KO and cosmotropicity is, because this is reflective of the salts I chose to use. If you look at the bottom right-hand side of the slide, you'll see a scale that is known as the Hofmeister series. On the very left-hand side of the Hofmeister series are very cosmotropic anions and cations. That means that these ions stabilize the hydrogen bonds also in the brine solution, and therefore also stabilize biomacromolecules that are also found in that solution. Proteins, amino acids, micelles, anything like that. When you go to the middle, from the far left to the middle, you get to sodium chloride. That's our neutral species, which we generally perceive as having neither KO or cosmotropic abilities. However, we'll talk about that later because I have some views on that. And then on the far right, you'll see the chaotropic ions, which destabilize the hydrogen bonds in the brine solution and therefore destabilize the biomacromolecules also in solution. And if you look at the top right-hand side of the slide, you'll see a bell curve, and the peak of this bell curve shifts based on the nature of the salt, whether it's KO or cosmotropic. So what we want to do in this project is more closely define KO cosmotropicity, because there are a lot of planetary dynamics related to the existence of salt in that planet, in an ocean world, or even on the surface, like on Mars. There's a lot of ways that salts can interact with a lot of different things. And so we want to know on a basic level how salts would affect life. And so that is what we decided to do here. And we opened the book into how we can learn more about habitability, bioavailability, and also figure out how planetary protection can more closely define special regions. So I just want to, I'm not going to do this whole graph, but I just want to let you guys know that the salts that I use are sodium chloride, my neutral analog, magnesium chloride, which is my chaotropic analog, and then ammonium bicarbonate, which is my cosmotropic analog. And these are all solar system relevant salts, especially on ice moons, ocean worlds, and Mars. Okay, this is my protein. This is called the prokaryotic ubiquitin protein. This is the precursor to eukaryotic ubiquitin. As you can see, it's intrinsically disordered, which means it has no secondary structure, which is why I chose it, because I wanted to see if my salts were able to add secondary structure or anything. I had no idea at first. I just wanted to get a blank slate so that way I would be able to clearly see any changes. And what we did is we exposed that protein to 0.15, 0.5, and 0.75 molarity of each of my salts. And of course, in these solutions, it's only salt and water. And I utilized VMD with the CHARM and NAMD GROMAX extensions to look at all this. And I ran my simulations for 160 nanoseconds, but then I extended them to 250 nanoseconds later. And those are the results that we're going to be also discussing today at 273 Kelvin at 1 atmosphere pressure. Okay, so these are my results. These molecular dynamic simulations take a long time to run. It took me a long time to get these. And this was really disappointing because there's not much change, right? So the strings on the left and right side, those are amino acid strings. And as you can see, there's some lengthening in those amino acid strings, especially in the 0.5 and 0.75 molarity, but there's no amazing structural change here. Moving on to sodium chloride. Again, not much structural change. You can see very intense lengthening of the amino acid string on the left side of the 0.5 molarity of sodium chloride. But again, they're pretty comparable to the unexposed PEP protein. However, when we exposed the PEP protein to ammonium bicarbonate, which is our cosmotropic anion-cation combination, we actually did get a folding event. And I have never seen that. Okay, I've been doing this for 3 years and I have never seen that. So when I saw that, I got extremely excited and decided to do more research. And you can also see there's kind of like a half fold at 0.75, but it's nothing like this. So this is actually what happened with my PEP protein. It takes 2 expenditures of Gibbs free energy to achieve folding, and you'll see the first partial fold here soon, where it tries to fold right here, and then the protein relaxes because it takes just as much energy to unfold as fold. So it's the second expenditure energy that allows this PEP protein to gain a folded structure. And he's just doing a little dance for you, so I'm just going to move on. Okay, so there's something really important that I mentioned about that folding event, that it occurs in 2 phases. And if we look at the RMSD, which is the root mean square deviation of my simulations, we actually get the 2 peaks on the top right, as you can see with ammonium bicarbonate in red. We get those 2 peaks at the 0.5 molarity, which is representative of that 2-phase folding event, which intrigued me even more. So then I decided to do a violin plot to get an idea of what I was seeing on a more grander scale. So I repeated all my simulations in triplicate, and then I extend them from 160 nanoseconds to 250 nanoseconds. And it gave me a better idea of what was going on because it gave me a really big spread of data. And if you check out the x-axis on the bottom, you'll see the first 3 are sodium chloride, which are our neutrals. Our second 3 are magnesium chloride, which is our chaotropes. And our last 3 is ammonium bicarbonate, which is our cosmotropes. Now, do you remember that bell curve I showed you earlier? If we move those 3 sodium chloride species to the middle to where it's like the Hofmeister series and goes chaotropic, neutral, cosmotropic, we get a bell curve. Very interesting, right? So I decided to look into this more. And the more I looked at it, the more I realized that, oh, first of all, I would like to describe to you what you're looking at, because not everybody knows what a violin plot is. The height of the violin is the deviance, the amount of deviance of our exposed PEP protein from the unexposed PEP protein. And the width is how often that was reached by that protein. And so as you can see, sodium chloride was able to reach a whole bunch of different conformations, just not very much and not very stable. And then we get to magnesium chloride, our chaotrope, which is kind of in the middle. There's more frequency, but not as many other diverse options. And then we get to our cosmotrope. Now, I want you guys to think back when I was talking about that 2 phases of protein folding. If you look at that 0.75 ammonium bicarbonate and that 0.5 ammonium bicarbonate and even the 0.15 ammonium bicarbonate, they have a bimodal distribution, which means they have these 2 bumps that appear, which is reflective of what we would see with the 2 phases of protein unfolding multiple times. So I wanted to see what happened. So I looked at all my proteins and I was actually able to find that not only did the chaotropic anion and cation force folding, but also the lower concentration of my neutral analog induced folding at the longer simulation of 250 nanoseconds. And there's also an appearance of a alpha helix at the bottom right. That's that little blue swirly thing. And I did a lot of research on that, and I found out that if a protein folds and is stable, maintains its fold and develops an alpha helix, this is representative of the official stable change from primary to secondary structure, which is very significant when we're looking for not just biosignatures, but the evolution of life and things becoming more complex, et cetera, et cetera. So just to wrap things up, I've got 40 seconds. I was able to look at all my outcomes to 250 nanoseconds, and the ammonium bicarbonates did remain folded 2 out of the 3 times. So to me, that's statistically significant, and it shows me I have more work to do. And it also gives an idea of which salts we need to protect in planetary protection in terms of habitability, all these things, because these special regions, these salts allow us to have special regions where these folding events can happen. And when proteins fold, I promise I'm almost done. When proteins fold, they open up in certain regions that will offer more biochemistry to happen, which makes things more complicated and more advanced, which is why we're here today. Thank you very much, and I'll answer any questions you have.
And if we could have all those speakers come back and we'll do our last Q&A.
Hi, David Bresse from University of Florida. For the last talk, why did you choose these concentrations, and did you try more restrict these ranges for the ammonia chloride to understand why it appeared at 0.5 molar and not like weirdly at 0.15 or 0.8?
Yes. So my current limitation now is supercomputing time, because it takes a long time for these molecular dynamic simulations to give me outputs. However, you're totally right. I'd rather do a millimolar concentration because that's more along the lines of like actual reality. But, you know, it reminds me about Catherine Zarilla's talk earlier when she was talking about, she kind of looked at extreme examples to get a clear idea of what was going on, and then she backscales in order to see like what happens like in reality. So that's probably what I'm going to wind up doing, because I want to see what's going on first and then like go from there. But what you're describing is absolutely my next step.
And like, even if you go to millimolar, so maybe it was not long enough, but you didn't observe anything at 0.15 molar. So did you try 0.4 and 0.6 to understand why it folded at 0.5?
So I have done a little bit of research into that, and what it seems to me is that there's a Goldilocks zone with these salts. So if there's not enough of the salt, then there's not enough of the salt to make a change. If there's too much of the salt, then it denatures the protein, and you lead to like salting in, salting out. And so that Goldilocks zone is really important, and it's also dependent on the nature of the salt. So I'm like playing with dice here, trying to figure out like what I'm looking at, like with a little microscope. But you're absolutely right. I do need to more closely monitor, and even to like a 0.01 difference exactly when I'm seeing that folding. And I would like to replicate that in the lab also with X-ray chromatography. That way I can also see with my own eyes what's really going on. Thank you.
Adis Husic from UT San Antonio. My question's for Joey. Sorry, Joey, I'm hiding behind the podium. So in the very end of your talk, you kind of showed how you're mixing the tholins with some organic compounds. And if you could maybe talk about that a little bit more, like, is that not surprising that you kind of get like this, you can see what you're mixing in, but you also see those tholins very clearly, or?
Yeah, good point. And just as a quick plug for Adis's poster, which is showing the new group of tholins from University of Texas, that's on Thursday night, right? Yeah, so HUSIC, HUSIC poster Thursday night. So yeah, I'm glad that I can see them. And to your point, like, was it obvious that I was going to be able to detect these prebiotic compounds when they're mixed with the tholins? And like, no, because the tholins produce so many ions that I was extremely worried that those ions were going to swamp out whatever kind of ionization potential is there for the prebiotic compounds. So I was afraid you were going to get a big grass of tholins and maybe like a hint of prebiotic compound in that spectra. And operating on Titan in mission relevant timescales, it's going to be really hard to find those compounds within that grass of tholins. So that's why part of my project is using machine learning to train to try to detect those prebiotic compounds as well. But it wasn't obvious until I did the experiments that the tholins weren't going to totally swamp out the prebiotic compounds. That was something that, like, once you see it, it's like breathe a sigh of relief, great, but it was not at all what I was afraid of. Yeah, good question.
And then if I can ask a follow-up question, how much actually did you mix in? Because you didn't state like the concentration.
Yeah, totally. So I did, so I optimized for LDMS analysis of both tholins and prebiotic compounds separately. So the tholin ratios, it was 2 per mils in methanol. And then I did that with a concentration of 100 millimolar of each of the prebiotic compounds. So that's the mixing ratio of 1:1 of 2 per mil tholins in with 100 millimolar prebiotic compounds. Next steps would be trying it with all the different tholin sets and then lowering that ratio of prebiotic compound to tholin to see where that cutoff would be for when you can still detect them. But this was first, like, in best case scenario, when I know I can get a signal from both types of compounds, can I still detect both? Yeah. Thanks.
We're going to go to an online question real quick, and then we'll return to the in-person questions. We have a question here for Denise. The question is, how does the life tracer model, excuse me, just a second. How does the life tracer model differentiate between biotic and abiotic organic distributions, and what are its potential applications for analyzing samples from Mars sample return missions?
Yeah, thank you for the question. Oh, whoops, what happened there? Okay, yeah, so basically what it does is it ingests the chromatographic information. So that includes a combination of the peak locations, their M/Z values, and the intensity of each of those ions. So yeah, it's a combination of kind of like the entire profile of organics as they're distributed across the entire sample, but you don't necessarily have to know what the identity of that molecule is. So it's really a powerful tool for analyzing a large set of like complex organic material where you might not necessarily know what every specific isomer is. And I think it could be used for identifying potential biosignatures in samples that were returned by MSR. If you have a lot more samples, we have only just started this project, so we have a set of like 18 samples, but we'd like to continue expanding, maybe using some Martian meteorites or other types of samples from Earth. And if we can start to understand how different populations of organics plot in different spaces, if we have samples that are brought back from Mars and we find that some of these organic distributions are plotting similar to one of the different populations that we've identified in our data sets we've curated here already, that could maybe give us an indication of what those distributions are more similar to.
Venkat Manga, University of Arizona. My questions are to the last speaker. My first question is about the statistics, and then the second question is about the mechanism by which such a folding could happen. So related to the statistics, can you try by adjusting some parameters like temperature or the concentration, by playing with some of these parameters in the calculations, can you get same probabilities for folding? That's my first question. And then mechanism, if you say, no, this is definitely related to these anions in the Hofmeister, in the series. So what is that mechanism that is actually enabling the folding with some anions, and how do you see that really characteristic of those species?
Okay, definitely. So I'd like to answer the first question first, which is, did I see the same thing with other temperatures and other like pH and things like that? So I actually chose a different protein called an ANCC ancestral protein. And this is like back to where the mesophilic bacteria first came into existence. And this protein has secondary structure, and it was not as easy to see the changes in the protein because there would be such strange things going on, like a beta sheet would disappear, but an alpha helix would appear on the other side. And so There are very specific reactions that happen based off the protein that you're looking at, that it's more, what you're talking about is more dependent on the protein you're looking at than like temperature or whatever. And obviously like temperature would matter, but I'm not going to extreme temperatures yet. I'm staying within like the same temperature range, 273 Kelvin. So that's like the biologic range where like VMD can actually still run simulations and not crash and things like that. If I am going to go into cryogenics, it'll probably have to be in the lab because simulations have a really rough time with like low temperature because there's not as much energy to do things. And so it struggles to do that. I have increased the temperature to 293 Kelvin and didn't see much of a difference, but that's not increasing it by much. I haven't played around with pH or other thermodynamic properties such as like water activity or anything like that. But if I incorporate additional software, I probably could. And I think especially if I changed pH, I think that I would get different results. And that will probably be like a complement to that I'll do later to what I'm doing now after I learn more about like the basics of KO and cosmotropicity. Now, what you're talking about with the protein folding. So what I've found out is that when you put a protein like PUP, which has very positively charged tips on the amino acid strings, it folds because there are electrons that are moving around and there's a lot of electronegativity there that does additional chemistry. And so there's a lot of stuff happening on the ends, on the terminal sides, and that kind of starts the folding event to happen. And when there's something called zipping and assembly that happens, where the internal hydrogens kind of like start going towards each other once the protein starts folding, because the hydrogen bonds are enhanced due to the presence of the cosmotropic salt. So as the protein is opening up on the terminal ends and coming together very well, it zips up and it stabilizes in that way because it's able to have those van der Waals bonds to like stabilize its folded form. And what's interesting about that, I'm glad you brought this up, is that as ubiquitin goes from prokaryotic to eukaryotic, it must fold. And so that shows that you can go from primary to secondary structure in an evolutionary way with just salt, because salt opens up those ends and opens up the inner sides as it starts to fold for more chemistry. Does that make sense? Awesome.
Thank you.
You're welcome. Thank you.
Sorry, I'd just like to jump in from online. For George Cooper, I apologize for having to cut you off. Did you want to take a minute or so just to recap your conclusions and take home message?
Oh, no, it's just these enantiomer. I was basically finished. Like I said, we just literally took these results off. Having deuterated water on interstellar grains, where you could substitute for hydrogen, you make something that wasn't chiral, glycoaldehyde. Now that carbon has 4 different groups. It doesn't mean it happened. The reason we get these enantiomer excesses of sugar derivatives in the meteorite doesn't mean that's the way it happened. But we could do it in the lab now, and I would bet the house that the chiral gymdiol, chiral glycoaldehyde. I have one paper where they were searching and looking at the spectrum in the lab. I don't know if they found chiral or deuterated glycoaldehyde yet on the grains, but I would bet the house that they are there. Bet the house. I rent the house, but it's the thought that counts. I would bet that they're there.
Awesome.
So
Thank you.
Bottom line, yeah.
Hey, just want to say really cool talks for everyone. I thoroughly enjoyed them. I would have a first question for you, Joey. If I understood well, you're trying to build a library of tholins to just make fingerprints of each one of them, right? And I'm just curious if you have like a specific parameter in mind that you want to explore with all the tholins, being like the energy source, the methane fraction or whatever, or are you just trying to explore everything that is possible? Or are you focused on a specific parameter?
Yeah, right now just exploring everything that's possible to then see what kind of differences even exist when keeping the same gas, and then just exploring different ways of making it to see what that's reflected like in the LDMS spectra.
Okay, okay, I
Yeah.
see.
Yeah.
And I would have a second question for you, Shelby. I thought your talk was very cool, actually. I would have a lot of questions, but I was just going to ask one because I don't want to take too much time. But just generally speaking, do you think it's possible to also see what you saw experimentally? And do you have a plan to do that, or?
Okay, awesome. I'm glad you asked this because I ran out of time because that weird thing that happened in the beginning. So I've been utilizing UV spectrophotometry. However, I'm uncovering some issues because a lot of the protein viewing techniques that we have only look at certain regions. And UV-Vis is a really good example because we only see that what's going on in the tryptophan regions. And so in the case of PUP, there are 2 tryptophan regions. And so it makes it kind of confusing for me to understand what's really going on in the lab because I'm not getting the full picture in the lab. But what I'm starting to find out is that I need to utilize hyperspec technology, which goes below 180 and above 450 to kind of give me a wider range of what's going on. And there's also like X-ray chromatography where I can actually like get a snapshot, but those are like all in the future. What I've done now, it's pretty ambiguous. And I'm not having the easiest time replicating this in the lab, just to be honest with you. However, I think once my research becomes more popular, I'll have access to more fancy equipment, and I'll be able to see more clearly what's going on with my proteins in the lab, especially if I can, how would I say it? Like use, like do a cryo stain on it.
What I was thinking of
Yeah.
IREM would be maybe a good idea, but I don't know if it's possible because of the size of the protein.
Right. You're exactly describing the issues I'm having in the lab right now. And I think I'll overcome those issues, but it's just going to take a lot of sensitivity assessment to figure out what I can actually do and what I need to do to actually get good results.
Would it be possible to do like FRET? If the protein folds, you could get like a transfer of fluorescence between 2, but maybe it would be too big and it would influence the folding.
Well, you're totally right. And that's what I want to do is use fluorescence. However, if the protein doesn't fold, we don't get much of a signal.
Yeah.
So that's the trade-off. And it's more likely that the protein will not fold than will fold. So my advisor is not like super into that, but I'm going to probably do it anyways, just to do it.
Sure.
Thank
you a
you.
lot.
We have another minute or two here that we do have an online question from Mireya Cortez. This one is for Joey. The question asks, does LDMS contribute to distinguishing between complex CHN compounds and prebiotic compounds on Titan? And what are its implications for detecting potential biosignatures?
Yeah, absolutely. So LDMS absolutely distinguishes between complex CHN compounds and prebiotic compounds. Implications for detecting, so the way Dragonfly is going to operate is LDMS is going to be the first survey mode for analysis. So the ability to separate out these prebiotic compounds from the complex CHN compounds is paramount for mission operations. So being able to make those distinctions between the complex CHN compounds and the prebiotic compounds when they're mixed together was kind of the whole point of the research, to make sure that we actually can effectively separate those different types of compounds out. And the implications for that for prebiotic chemistry are, huge. So but that's why that LDMS was selected, is because it has the ability to do that type of analysis. I'm demonstrating that what was hypothesized to work actually does work.
I'd like to ask one last question before we wrap up. This one's for George. George, you had mentioned at one point in your slides that you had previously seen a large D excess for threonic, but no coinciding excess for arabinoic or erythronic. And you postulated that if the large D excess for threonic were to be due to terrestrial contamination, you would also see a large D excess for arabinoic and erythronic. Do you think that it's possible that there could be a potential point source of contamination for one species over the other, or would you necessarily expect more of a uniform contamination if contamination was actually occurring?
Well, that huge excess in D mirror image for threonic, you always see the corresponding reverse when you reverse your magnetic field. Now, your point, there were excesses in the other. It's just that if that big D mirror image excess was a fluke contamination, then you should have had a lot more erythros, erythronic, or arabinose, whatever their mirror image was, because again, you accidentally drop something in your test tube or have fungus contaminating your samples. What do you see? Glucose, erythros, arabinose, and no, the threonic is in nature, but it's nowhere near. I've never seen that contamination profile in anything. I've looked at soil. So I was saying that D, it was last year was such a huge excess that maybe it's some kind of fluke. But now that we discovered the trick is maybe all of this deuterium on interstellar grains making even formaldehyde chiral, or its main form, then that goes back to maybe that wasn't a fluke. We can't explain it by contamination. Where's your glucose contamination? So the others are fine. Their excesses were in line. It's just that those experiments is so hard. It was so hard with C12, C13. You would have to do 10 experiments before you saw the excesses again. But now, back to back to back to back. So now we could publish the thing. So, so the others were fine, Rabenos.
Great. Yeah, that's very interesting. Thank you, sir.
Yeah. And I wanted to talk to you about your talk, but budget rental car messed that up the other day.
We'll do that right after the session here. Thank you. All right, so that'll wrap up our session for today. Thank you, everybody, for attending. Thank you to all of our speakers. Let's give them a quick round of applause. And once again, if you are able to serve as a Dwornik judge, please do sign up and give our students some support and some feedback. They've worked very hard for it. And there is also a virtual poster session tonight, if you're able to attend that. And there's an in-person poster session on Thursday. Both of those are relevant for this session. So thank you again, everybody, and we'll see you at the poster session.